at what we are doing in this region of the world, that is in terms of nanotechnology in health, that is what are the specificities of these different labs and how we could put this together and contribute more to the Southern African region and the Indian Ocean region, of course. And we have the WITS Advanced Drug Delivery Platform researchers with us. We have SIWA, which is also a research platform, and we have the CBBR from the University of Mauritius. And just to give to you a brief overview of our program will stand as this, that is, we will be having a slot of 15 minutes, and then we'll be taking the questions at the end. And we will leave also the floor open for new things that we could have in the African region. Nanotechnology, we usually say that it's the technology of the tiny. And some people sometimes think that nanotechnology is just the future, but nanotechnology is not the future. It is more things will come, but it is already the present. And I've just taken the case of the current COVID-19 pandemic situation. And there are lots of things that have been done and that we are using. We've got the vaccines that are already being used using nano-based technologies. We've got different types of respiratory masks being used, air filtration system. And it was very nice to see that in hospitals, some hospitals, we are using nanomembranes in the air filtration systems. We've got disinfectants, medical supplies, so many things coming from nano-based technologies are already being used for the COVID-19 situation. Now, for this to be possible, we have to be ready. That is, all these labs, countries, we already need to have the basic structure to be able to use nano-based technologies in any situation that can crop up. And I analyzed the nanotechnology progression in Africa. That is, at the very beginning, whatever was happening in terms of nanotechnology was basically driven by principal investigators. That is, this PI, they set up labs, and then from that we moved to really African-based nanotechnology labs. And years of now, we are seeing continental movement with AMRS, that is the African Materials Research Society. We've got UNICA, we've got the African Union. This is the direction that nanotechnology is taking in the African continent. And for today, we have basically people in the Southern African region, the Indian Ocean region. And we are also doing this under the umbrella of UNICA and the African Materials Research Society. And we are hoping that the different regions on the African continent could organize themselves in hubs, and then we could plug in the inputs of all these hubs together. And this is just a first input to that. And I would say that since last year, a lot has been done with AMRS and UNICA on the African continent to promote the nanotech field. And this is also in line with this. And I would like now Professor Yaya, who is the director of the WITS Advanced Drug Delivery Platform, to say a few words of introduction to this webinar. Yaya, to you.
Thank you, Archana, and thank you, participants, and the opportunity for us to meet virtually this morning, and obviously to highlight some of the work that we're doing in the southern region of Africa in terms of nanomedicine research. And also the focus being nanomedicine, as Archana also pointed out, nanotechnology being much broader, obviously. And also to highlight where is the, the promise of nanotechnology and nanomedicine and the future of nanomedicine for the African continent and for the world as well. You know, when nanotechnology hit the scene and nanomedicine hit the scene, we all, the scientists, were very excited, that we're going to revolutionize the way healthcare is going to happen. And in some instances, I think people overpromised, and in some instances, there have been issues in terms of challenges of getting translational research to the market, where nanotechnology can be at the bedside, helping patients, etc. And I think we need to obviously revisit some of the way we do things on the continent, so that these pockets of excellence that are available, how do we harness communication between various research groups? how do we fund and what models do we use to fund researchers? It should we use the big, a large research group kind of funding model, or should we look at these pockets of excellence and see where we can direct funding so that we can build capacity in these areas and then virtually, obviously, create systems and networks for people to have access to facilities, equipment, training, and the regulatory environment for these nanotech research products to be made actual clinical products for impact. And I think that's where we are focusing as well. In terms of the training and capacity development, I think there's lots of good things happening, but not much being coordinated at a level where we see the impact actually coming out into the marketplace where the impact will be made. And I think that's one of the things that we're looking to achieve in this, I will say, community of practice, for this, for lack of better phrase for now. But I think it's exciting. I have a team of researchers with me joining us as well today from the VITS Advanced Drug Delivery Platform, and we look forward to a positive engagement. Thank you, Archana. Thank you.
Thank you. And now I'll pass on to Samuel, who is from the African Materials Research Society. And he's going to say a few words about what we are doing in terms of nanotechnology, the vision for Africa. Samuel, the floor is yours.
Yeah, thank you, Achana, for the introduction. My name is Samuel Chigome. I'm the president of the African Materials Research Society. So for this session, I'll say some introductory words together with my colleague from UNECA, Vita Conde. I think you'll see him shortly, he just joined. So I think what I can just start off by saying is that as African MRS, our overall objective is to promote excellence in all aspects of materials research. So nanotechnology is something that is in line with our main objective at MRS. So as you know that it's important that we always partner and work together as teams for a common goal. We got into a program cooperation agreement in the last quarter of 2020 with the United Nations Economic Commission of Africa for the promotion of nanotechnology in Africa. So this half-day symposium that we have today is also in line with some of the activities that we are having with UNECA. So I think the way we look at nanotech for African development, we can look at it from the point of view that nanotech in principle, we are not necessarily saying that we are doing something completely new, but we are, from the African perspective, is an opportunity to take advantage of the abundant resources that we already have. So I just refer you to Richard Feynman some time back, where he talked about having plenty of room at the bottom, where all of a sudden, because of the knowledge of nanotech, there are a lot of opportunities that he initially didn't know that they were there. But then because of nanotech, there are a lot of things that can possibly open up. So I think we're looking at it from that point of view, that let's make use of nanotech to add value to what we already have and develop our continent. So that's the framework in which we're looking at this. So in terms of how we can go about it, we are looking at it from the point of view that as much as nanotech is applicable in a number of areas, almost every sector, probably maybe the best way is to look at it in phases. So we are looking at it from the point of view that we can have maybe 4 initial focus areas that are nanotech for health, agriculture, water, and energy. And then we see how that develops before we then expand to go to other areas of nanotech. So in terms of how we do that, I'll just adopt this slide that I got from Professor Maza. He's a board member of MRS, and then he's also the UNESCO UNISA Africa Chair. We are looking at how we can look at people that are already involved in nanotech, the research labs that are already involved in nanotech in Africa, the different things that they're focusing on. And then we can see how we can maybe get them involved probably in initially maybe the 4 focus areas of nanotech, because obviously for us to be able to develop, we need to have people that are on the ground, that have some work that is actually going on. So I think you can already see that University of Mauritius is part of this network of nanotech institutes. Bitri is also there, although we're not going to talk about Bitri today. But then I think we also have VITS that is involved that's not really showing up there, but we also want it to be part of this network. As we have today, we're talking about nanotech for health. So I think the idea that is that we want to find a way of bringing together all the units in Africa that are looking at the different aspects of nanotech, so that we can be able to work together as Team Africa. So that's what we're already working on. So in terms of what this will translate to, looking at the initiatives where we've brought together the nanotech nodes, we have, I can say maybe about 5 sort of initiatives that we have. We have I can refer to them as nanotech bootcamps, or these are just what we normally refer to as the nanotech summer schools. So what we do with this is that we understand that it's important to be able to develop students because they're the future. So the idea that we have with this is that we create an environment where we can maybe say have about 100 students, 20 from each of the 5 regions of Africa. So they come together maybe for about a week. And then what we then do is that we look at how we can have world experts in the different aspects of nanotech that can also share some of the work that they've been doing. And then that creates an environment for students to get exposed. And then it helps to sort of energize them and create an environment for mentorship of some of the ideas that they have. So what we expect from this is that it should allow us to be able to identify some talents. Let's say maybe where we have a series of bootcamps, remember about 5 of them. If we select maybe 5 students from each, then eventually we can then get the students selected to be attached to the different maybe nanotech institutes or the different world experts. And then from there, they can serve as their mentors, which would then allow them to develop and then eventually either set up their own groups or maybe eventually set up a company in Africa. So this is something that we can see as a sustainable way of developing people going forward. So it's something that we're looking at implementing as one of the initiatives. And then the other thing that we're also looking at is how we can promote research that is focused on Africa in nanotech. So we are working on how we can have a Pan-African MSc and maybe PhDs in nanotech that can ensure that going forward people do research that is focused on solving some African challenges that we have. The other thing that we also have that we are currently doing is looking at webinars and workshops. For example, like today we are having this afternoon symposium on nanotech for health. So what will happen is that it helps to communicate what the different labs are doing, because as I think it was introduced by Achana and Yaya in the beginning, that one of the challenges that we have is that people don't know what we actually have. So this is sort of information sharing that helps to make sure that at least everyone is aware of the facilities that people, different people have got, and also their research directions. That way, it ensures that we can be able to team up and then work together as a continent. The other things that we're also looking at doing is that as much as COVID has restricted the movements, but in a way, I think it has sort of helped us to realize the great potential that we have through doing things online. So we're also looking at how we can have online demonstration workshops or maybe virtual labs that we can actually run, where we can even have online demonstrations where we can show some experimental work that can allow students in different aspects of nanotech to actually learn some experimental techniques. So we're looking at how we can organize some of these in the different areas. So I think if we progressively do these things, they will help to ensure that at least people have the requisite skills for them to be able to develop in the different areas of nanotech. The other aspect that we're also looking at is that a way of making sure that we move away from having nanotech as just being just a research, should I say, aspect of our lives, where we just look at it as you do research, you publish, but then we also want to look at it from the point of view that people should think in terms of entrepreneurship. People should think in terms of industry creation. People should think in terms of employment creation through nanotech. So this is something that we're also looking at how we can be able to sort of instill this way of thinking in nanotech. So we also have the different initiatives where we look at, for example, like if I can talk about someone like Professor Sabelo Mhlanga, he's got a company that came up from his work on the synthesis of carbon nanomaterials. Is currently stationed in South Africa at Mintech. So we also have another company by Eugene Smith. It focuses on electrospinning, where he's looking at industrial scale electrospinning. So all these are some of the examples that we have of companies that have actually come up from nanotech. All these were researchers from universities. They were spin-offs for universities. Now they are companies that stand on their own. So we're looking at how people can actually see that we can have all these companies that we can actually have that can come up from nanotech. So we also want to also move forward and promote the entrepreneurship aspect of nanotech. And then I think looking at all these things, what we are looking at going forward is that we want to be able to get to a stage where we develop a Africa-wide strategy for nanotech that can guide all the activities that we have. But for example, like now I talked about, initially talking about nanotech for those 4 areas. But then I think eventually what we would want to do, we would want to get to a stage where we talk about the different regions. If sometimes we may find that we may have regional challenges that we need to be focused on. So we want people to be able to also look at those things as well. And then I think some of the things that we also want to also make sure that they're in place. If you look at the nanotech policies for the different countries, where that can also be captured as well. So I think in summary, this is what I can just say, that that is what we look at from our perspective, how we can be able to move forward in terms of realizing the vision of nanotech in Africa to develop our continent. Thank you. I think I'll just stop from there and allow my colleague Victor to probably just take over from me. Thank you, Ashana.
Well, good morning, everyone, and I hope you can hear me. My name is Victor Konde, and also I think my director actually did join us. I think he's still with us, Jean-Paul, with the director of the division in which science and technologies is hosted. And I think has been a major supporter for the work that we're doing. So it is good that he has joined us this morning. So perhaps just to maybe just to say a few words among the issues perhaps we need to focus on anyway on behalf of is perhaps we need to prioritize, because I mean nanotechnology can be applied everywhere, but we need to figure out on where do we start and then where are we going. I mean the decade we are in is what everyone is calling the decade of action. And if we're going to take action, we need to agree on a few things that maybe we can help our member states to focus on. Now, agreeably, the traditional sectors may be important. In some countries, perhaps issues around health are common to all of us. But there are countries in which, for example, health is perhaps big, where we all run to when we are sick. And in those countries, perhaps the health sector is well developed. They have the industries, they have the regulatory environment, and they have the investment infrastructure already in place. And therefore, there is actually a dynamic health sector that they can work with in promoting, for example, nanotechnology. In other countries, for example, issues to do with mining or issues to do with maybe agriculture are also well developed, in which case the investment environment does exist. The business environment actually perhaps is already well set up. The regulatory issues are in place, and the basic infrastructure, for example, may already be in place. And COVID has actually taught us that. We saw a number of minds actually reach out to, for example, the universities and research centers in helping them to cope with how do they go out to quickly test. So whilst maybe the countries were doing testing using the PCR and taking maybe 2 days to give you results, at some of the mines actually they could tell you their workers immediately within maybe a couple of hours that yeah, we have someone who is infected. Remember, we are talking in the general community, of course, there are people at the space, but in a mine, actually people literally work together and they are going to go underground, for example, in many of our countries with underground mines. So I'm thinking when we are looking at nanotech for a number of countries, we need to, there may be a place where we can maybe talk to policymakers and say, this may be where we can start. Maybe it may be in agriculture, it may be in health, or it may be in mining. And so from our perspective, when we look at the continent, I think if we pay a little bit more attention, we should be able to have maybe a little private prioritization. Of course, the issues of human capital development may be very important for the continent. I think my colleague Samuel may have not mentioned it, but one of the reasons we are asking everyone that we should do the boot camps is actually to build up that human capital base. Because whether we are going to promote, for example, innovation, or we're going to promote business development, or we will need actually the manpower, the necessary manpower that will actually bring these solutions to market. And also for these are knowledge intensive. More importantly, of course, it's a very interdisciplinary field. So we need people not just who are specialized in chemistry, not just specialized in engineering, but we may need people who are specialized maybe in computer science or in mathematics. That may help, may find different uses of nanotechnology, like you said, in different areas. So from our perspective, this is one of the reasons why the work that we are doing at the United Nations Economic Commission for Africa on nanotechnology is really part of this bundle of what everyone's calling emerging technologies. The one that we are hoping is going to push Africa into the fourth industrial revolution. And therefore, from that perspective, I'm hoping that this is perhaps one of our learning maybe one of the lessons that we'll bring to our member states and some of the support that we could provide. And we are counting on all of you to help us in doing that and also in helping our countries. For example, how do we build up the best business environment? Because there's no point in asking people, okay, go start up a business if they actually can't really start a business in their country, or starting a business in the country takes so long. And that may not necessarily be nanotech, but it will affect all the other businesses. But then it will affect perhaps nanotech more if, for example, I mean, we will have learned, for example, in during the COVID-19, just trying to develop a ventilator takes you more than 2 years. And this is a basic, and then the people you're trying to help are sick now. They are not sick waiting for 2 years to happen. So in a way, there are a lot of things that we may have to do together. And so I'm hoping that we can all learn, and I'm glad that this time we are focused on health. And I think health is important to all of us, and the lessons from health are likely to have spillover effects in the other fields. So I'm happy to join. I don't think you came to listen to me, but I think we have a number of interesting subjects that we need to address this morning. So I'll end right here. And thank you very much for joining us. And thank you very much for partnering with ECA. We look forward to working together this year. We still have quite a couple of months to go. So there's a lot we can do. I'm not sure if I get John Paul around, but at least he can say hi.
Thank you very much, Victor, for introducing me, and just good morning to everyone. And I'm very pleased that we are able to move in this direction. I particularly like the idea of moving towards regional hubs, which can help us build capacity across Africa to then really link nanotechnology to the needs that we have across the continent. And our division within ECA is, some people see it as a strange mix of portfolios because it's technology, climate change, and natural resources. But I see it as as fitting quite well together, particularly in the context of sustainable development, where we need to harness technology to be able to really make sustainable development a reality. And I think this is particularly the case in the health sector, where we can hopefully give African countries the right technological know-how so that they can be less subject to some of the impacts that we've seen under the recent COVID-19 crisis, where distribution networks have been disrupted. The ability of African countries to make use of technology has been shown to be limited in many cases, because whenever there's a delay in supply, then our own technologies are disrupted. And this means from the outset, applying technologies that are, even if it's using technologies that are invented elsewhere, but applying them in a context which is very relevant for our countries. So I think that what we hope to see is really practical proposals, as well as practical proposals on how we can embrace nanotechnology to address the issues of sustainable development, particularly in health, and also ensure that we have the right ecosystem for the development of nanotechnology across, for example, the business sectors, because that will really be the way to drive it forward. So it's not my intention to speak for long. I'm more in listening mode. Thank you very much, and it's great to be with you all once again.
Thank you very much. Thank you, Victor. Thank you, Jean-Paul. Thank you very much for with this. We will make a start, and I'm going to start this webinar. That is, I'm going to give to you an overview of what we are doing on the African continent in terms at CBBR in terms of biomaterials engineering. That is, right now you will be having three centers. That is the CBBR, there is the WITS Advanced Drug Delivery Platform, and there's the CIWA Center. Each one of us, I'm very happy to hear Victor and Jean-Paul both talk about what we can contribute and how we can bring all this together. And each one of us, we have our different specificities, and this is what we want to bring together. And at CBBR, we very much take into consideration the SDGs in the things that we do. And just to give to you an overview of our activities, that is our research and innovation thrust, in fact, links our intellectual capital building and our service to industry. And whatever we are developing, we try to bring it to the society, to the community. And in doing that, we focus on SDG 4, 9, and 17. And partnership is what it is all about. And this is how we have been progressing in what we are doing. Therefore, today, I'm just going to give to you a brief overview of what we've been doing in terms of biomaterials engineering at CBBR. And just to give to you a small snapshot of what is going on in terms of the biomaterials market. In fact, the major factors driving the market is not only applicable to Africa and the Middle East, it is applicable to many other regions of the world. That is, biomaterials, their superior properties is what is going to drive the market. And also we have the problem of increasing aging population that biomaterials can be used to address. Now, in terms of market segment, that is where are biomaterials used, we've got, in health, we've got the orthopedics where it is greatly used. We've got cardiology, we've got plastic surgery, wound healing, drug delivery, and this is what Wits is going to talk about later. And then if we want to classify the materials, we have three types of materials that comes into this market. They are either bioinert, bioresorbable, or bioactive. And within this, we have ceramics, metallic materials, polymers, and natural biomaterials. And this is where we are working. We are looking at natural biomaterials because we are looking also at sustainability. And how did we get into that area? And our main hypothesis was that natural biomaterials, they could mimic human tissues, particularly the polysaccharides and proteins in that human tissue. And we were basically working in the polymer chemistry field. So we were already looking at all these properties of different types of material. And after 10 years, then we stepped into using natural biomaterials, and we engineer them. And we do three things with them. The wound healing scaffold is something that we've been working for the past 10 years. And we've just stepped into using biomaterials to make 3D tumor models so that you can study the behavior of tumors. And we are looking at nanofiltration membrane. This one, it really came because of the COVID-19 pandemic last year. And this forced us to rethink of what are the new things we could do with the technologies that we are developing to benefit the African continent. And why land and marine resources? This is where our biomaterials they come from. Our land and marine resources, really our marine resources is quite huge. Mauritius is here, Réunion Island is here. So you can see why the collaboration developed with Réunion Island. We have a huge space of marine resources available to us. This is our marine zone that we can exploit. We share this part with Seychelles here. It is a joint management area, and then we have the region around the Chagos archipelago. Therefore, we have huge marine resources that we could exploit. I'm going to show to you briefly what type of biomaterials we are looking at. We have been looking at seaweeds and algae as our main biomaterials because you can also grow them. That is, we are not just taking them from the ocean, but they can also be grown. And we've looked at green seaweeds, red seaweeds, brown seaweed, green seaweeds, because it was basically considered as a waste material. But this waste material has very important biomaterials there that can be exploited. And why collagen? Because collagen in the Indian Ocean region, there's a huge fishing industry. You've got the tuna fishing industry, and it produces a lot of waste. Therefore, you have from the skin and tail of the tuna fish, you can also extract collagen, which is a very valuable material for nanotechnology in health. And then something new also, which we have started with our collaborator, the Mauritius Oceanographic Institute. They've been working on marine sponges to isolate unique molecules in this region. And we are using with them the bioactive molecules, and we can put them into tissue engineering scaffolds to look at different effects. For example, they have got unique anti-cancer molecules, and we are trying to load them in nano-based devices and to study the effect on different types of tissues. And this is about an overview of the marine biomaterials that we are working on. And in terms of land resources, there are also a number of things that we are able to use from our land resources. The main one being cellulose from bagasse. Bagasse is at present being used to generate energy. It's considered as a renewable source of energy in Mauritius. And we can also use it to get value-added materials and sugar. From sugar, later Itisha is going to talk about maybe the work that she's been doing for a PhD. We can make very nice nano-based devices using sucrose as our basic materials. And what do we do with all these biomaterials? We have to understand the chemistry. And this is where it all started, in fact, understanding the chemistry. We blend all these biomaterials, we engineer these materials to produce scaffolds. And scaffolds, as you can see here, it is a support for growth. And we have made different types of architecture, in fact. And later you will see why do we want to do different types of architecture. It's because in the body, we see different types of tissues, different types of cells. Therefore, we make those different support for cell growth. And as you can see, we have the simple nanofiber scaffold that we make. We have the hydrogel scaffolds that we make. And in the middle, we have both nanofibers and hydrogel together to make hybrid scaffold. And this is something which we are working on right now. We are trying to bring the good properties of each one of them in a simple scaffold. Now, why do we do all that? It's because now we need to understand the biology. Once we understand the physical-chemical properties of the biomaterials, we engineer them into different types of architecture. Now we have to see what is the impact on the cells, what is the impact on the tissue, and we want to influence that cell material interaction because we are looking at wound healing in terms of skin and bone. And these are some of the pictures that we have taken of different types of cells involved in different steps of our wound healing process. And we look at the behavior on different types of architecture. We also put them together. For example, here you have different types of cells, and you can see how they interact with each other. They form layers. They interact, they talk to each other on these nanofiber scaffolds. Here you have a bone tissue, and you can see that this bone tissue is very happy on the scaffolds that it is on, and it's going to proliferate and do the process that is required for healing. And we also look at, here I've shown to you a picture of the same cell. This is a fibroblast cell, cells present in the skin. In fact, fibroblast cells are present everywhere. They are important for many processes. They are involved in cancer. They are involved in wound healing, in bone regeneration. They are everywhere. And when we put them on one type of scaffold, this is how they look like. These are the nanofiber scaffold. And we put them on hydrogels, they change the way that they behave. And these are the things that we are looking at that we are studying, starting from biomaterials. We understand the chemistry, we move to the biology. Now, what do we do with all this knowledge? What do we do with all the people that we have trained on these types of experiments, this type of expertise? We need to translate that, and we want to translate it in such a way that the African continent is going to benefit from that. That is why the focus on our local, regional biomaterials was very important from the beginning. That is, we need to have the raw material if we want really the region to benefit from that. And this is where our collaboration with CIWA, in fact, this collaboration started a long time back, and since 2011 when the center was created. And all this time it was just about building capacity and networks, and we were focusing on SDG 9 and 17 when we moved from biomaterials, doing architecture engineering, in vitro biology, and now into in vivo translation. Now, what new are we bringing to that area? With Siwa, Fanny is going to talk later during the day. We've been looking at small animal models for biocompatibility testing, wound healing in vivo testing, diabetic wound healing, and we've just started on in vivo bone regeneration. If you look at this picture, these are the wounds where we put our scaffold, and this one is one of our hydrogel scaffold, and really we're looking at scarless wound healing. These are you have commercial products that we have used. You can see scars present in them. Whilst this one is given to us, this is our own nano-based device, and it's giving a scarless wound healing. And we have more things to do on that. We are looking at the quality of the tissue, which is being formed, and so on. And then next, once we have studied all this, we, through the university, when the Vice Chancellor went to visit a number of groups in Australia, we were linked up with the ECU group, Edith Cowan University, the group of Professor Melziman. She works on the cancer. She's well known in that area. And then we started, we thought that we could use these scaffolds to model tumor growth, so that people that are working with cancer, they have a tool now to study really how these tumor cells grow in the 3D in vivo tumor environment. And this is just to show to you, these are the same cells. This is the cell when you're using a normal cell culture flask, plastic. This is how it's going to look like. You put it on a nanofibrous mat, you can see the behavior is already changing. You put it on a 3D scaffold, it completely changes its behavior. And we are not only putting one type of cell, we are putting different types of cells, which should be present in the normal tumor environment in the human body. Therefore, this is something new we are working on, using biomaterials, using the technology we have developed for wound healing. And since we've been looking at physical-chemical properties for years, we've got a lot of data that has accumulated, and we have just finished and published the preliminary work on modeling the scaffold performance using AI, that is predictive modeling, so that now we can have a tool, a model that can predict to us if we put biomaterial X with biomaterial Y, because at the beginning I told you that we are mixing biomaterials. If we put these biomaterials together, what type of behavior can we predict in vitro and eventually in vivo? The first preliminary work has been published, and we are continuing work on that, using, in fact, the chemistry to try and predict what should be happening when we translate. And the last one, which really the idea came forward because of the COVID-19 situation, where we got into developing nanomembranes, because I've said that we've been looking at nanofiber scaffolds for wound healing, but these could be used for other things. And in fact, we are really looking at biomaterials engineering to have a socio-economic impact. And we have developed a new type of membrane where you have the nano-netting phenomena. That is, you can use this membrane to capture bacteria and virus, and you can use it as filtration membrane. And this is a collaboration that we have developed with industry, because they make the mask and we make the nano membrane, and they are also quite well versed with the engineering part in choosing the correct structure for the mask, and we are working together on this. And this work in fact has allow us to do other projects with this particular industry, and we are having new projects coming out of these biomaterials engineering for socio-economic impact. Now, to finish, I would like to say that all this would not be possible if we do not have networks, and if we do not have a strong team. That is, this is the team that has been working. Of course, some of the PhDs, they have gone to do other things, but the core of the team has stayed behind as postdoc, as research fellows. We want them to build the expertise so that we can continue building on what we have done. We have our collaborators at CIWA, at ECU, MOI, Artinate, and of course our funding agency. And recently we received a grant from the EU to get our semi-industrial nanospinner. The equipment is in the lab, and we are all waiting to be able to use that equipment, in fact, for the nanomembrane to go commercial. And with this, I thank you for your attention, and we will be taking questions at the end. And I would like to introduce to you Professor Yaya, who is the director of the WITS Advanced Drug Delivery Platform. Here, I would like to mention that, in fact, it all started with the friendship between the two groups, which started way back with Professor Jerry and Professor Vines Pilet. And I think it's good to mention Professor Pilet at this moment in time. And I'm happy that this collaboration goes on with Yaya taking over. Yaya, up to you.
Thank you, Archana. Thank you to the UNECA team as well, colleagues as well as attendees and participants on this webinar. We are very excited to be part of this, obviously, and to create this platform to share with you some of the work we are doing at the WITS Advanced Drug Delivery Platform. Allow me to share my screen with you. I'm going to turn off my video as I start presenting, just to save some bandwidth on our side. Just bear with me for a second. Okay, thank you, colleagues. I'm sure I'm audible. You could hear me. So the overview of the nanomedicine research at the WITS Advanced Drug Delivery Platform. I also want to just pick up on what was mentioned earlier, by Mr. Conde, that this is the time for action in Africa in nanomedicine. Nanomedicine has been around for a while now. We were hoping to obviously see how it's going to revolutionize the space in nanomedicine and the health, especially in nanotechnology. And I think it's now time for action to see what is the bottlenecks and the challenges and how the way forward of how we can realize the impact of nanotechnology in its full sense. I mean, there's a lot of industries that have been forward in this in terms of engineering, and examples that are around there in terms of companies that have been set up. But in the nanomedicine space, there are still certain things that we need as scientists as well as commercial heads, etc., to get around and how do we actually make that impact. And I think platforms such as these, where the communication can happen, the training and capacity development issues that are really at the forefront that needs to take place. And that's how we could actually drive this nanotech agenda, especially nanomedicine in Africa. So just to go for an overview of nanomedicine research at the WITS Advanced Drug Delivery Platform, I'm the current chair and head and the director of the unit. As Archana has mentioned to you, the unit was started by Professor Vines Pilet, who sadly lost in last year, July 2020, who passed on. We were obviously taking his legacy forward is at the foremost of our mind, and my team who has joined us here today, as well as other members who could not make it to the webinar. And I also want to share with you some of the work that we are doing in nanomedicine research at the unit. So the WITS Advanced Drug Delivery Platform. So at the WADDP, as we know it, we keep commercializable nano-enabled pharmaceutical technologies in the forefront of our R&D strategy. And we do this having this commercial angle in our minds is because we know the impact of research can be translated to the clinics. And that's what we're looking at. Research has its place for the library shelves, but I think there's also definitely a research space that needs to happen in terms of translating the good work that we all do into the marketplace to make an impact on patients, impact on societies, communities, and obviously all the macroeconomic benefits that can be realized from research that has a commercializable angle to it. And we do this also to provide the pharma and biotech companies with the tools that to deliver their bioactives. Many times, pharma and biotech companies are looking for the next breakthrough, looking for, focusing beyond the generics market, the innovation market. How do we innovate? And we are looking at research that can provide the tools to provide solutions to the challenges that we have with delivering therapeutic molecules into the body, or to have the next therapeutic breakthroughs. And these are mainly for molecules that are difficult or impossible to deliver, and thereby offer more effective therapeutic solutions. So in a nutshell, that's our focus. The unit was launched in 2007. As you'll see, the name implied is VIT, Advanced Drug Delivery. So we started off as a drug delivery unit, and since then has grown to include lots of other therapeutics research areas, which includes biomaterials, nano and regenerative medicines, tissue engineering, and most recently 3D bioprinting, or what we term 3D bioplotting research and innovation. So as time has moved on, the complexity of the research space has also, we've moved with that as well. And now the WAGDP encompasses a quite broader range of the therapeutic space. It is a foundation for innovative research. We've trained more than 100 postgraduates, 60% are in the nano space, and also postdocs and nanoscientists. And just to mention also, majority of these postdocs are from the African continent. And that's why we see the value in obviously networking across the continent, and with these kind of platforms and programs and communication between the various institutes, so that we could drive training capacity, because that is one of the bottlenecks. It was mentioned earlier that you can start a company, you can have a brilliant technology, but you need people, and people are central to driving that impact. And the training and capacity development of true nanoscientists on the continent, of pharmaceutical scientists, is what is actually required to drive the nanomedicine agenda. We are aligned to our government strategy on nanotechnology, and by extension, obviously the continent, because we are part of the continent, there's no doubt about that. And therefore we are aligned to these strategies to make sure that, nanotechnology, nanomedicine gets to where we need it to be. We have a position of global competitiveness with a portfolio of patents that's more than 43 inventions. Some of them, 24 of them have been granted in international jurisdictions, and 30% of these are in the nanomedicine space. It's important that we obviously showcase, the pockets of excellence that I spoke about in my introduction, is that these pockets of excellence are all over the African continent, and we need to find a network or a home to actually communicate these pockets of excellence and translate them into what is truly needed, which is the impact at the bedside when it comes to nanomedicine, the patient bedside. It's focused also on improving quality of life of patients, and that's where you say by driving the business of science via academia or research institutes to develop new intellectual property, and also offer services to the pharma companies as well as the biotech companies or other research institutes that are driving nanomedicines research. So in a nutshell, that's basically where we started and where we've grown. In this slide, just some of our nano constructs that we developed in our labs. So we're working on my presentation here is basically kind of a graphical abstract that I want to share with the audience. I have two brilliant researchers who are joined this webinar as well, Professor Pradeep Kumar and Professor Lisa Dutoy, who will share some specifics on the details of some certain projects that we are underway. But in a nutshell, as a graphical abstract, we are involved in the development of like nano liposhells, nano wires, nano tubes, fibers, nanospheres, nano bubbles, nanomembranes, nanosomes, and all of these are packaged into different therapeutic modalities for different various disease conditions, illnesses, a bit of also on wound healing, tissue engineering, as well as infectious diseases for the African continent. So we can manipulate a nanoparticle or a nanostructure. This is just in the shape of a particle, but this could be a nanofiber, it could be a nanowire, it could be a nanobubble. These particles, we can manipulate it in terms of composition, in terms of adding drugs, in terms of changing the shape, as well as adding targeting moieties and ligands to them. So we have capability in all of these areas in our lab. I've included a picture here just showing you some of the equipment that we have, and also to mention, my colleagues will reiterate this when they do their presentations, but some of the equipment we have are first in the world in terms of 3D bioprinting platforms and tissue engineering. And we need to highlight this and see how we could help and also build capacity and networks within the African continent to make sure that these pockets of excellence don't get lost in the system. If you look at our scientific trajectory at WADEP, we started off our nanomedicine research at the organ level. If I take you to the bottom of this arrow that you see over here, and there we started at the organ level looking at nanoformulation. So basic nanoformulations in drug delivery, where our focus was on diseases such as TB, HIV, malaria, and now tropical diseases as well, where we looked at communicable and non-communicable diseases. And basically the focus there was to take drug molecules and see how we can improve the bioavailability, their solubility, and drug transport across organs. And the organs there mainly being the GIT, or the gastrointestinal tract, and how we can get oral drug delivery systems moving ahead. As time went on, we move on to more complex systems, and that's why this continuum I share with the audience. As the research area changes and the field develops, globally, as well as what's the needs for Africa, we have moved on to different levels. So the organ level research still continues in the background, and then we moved on to the tissue level, where we look at now getting these nanostructures across biological membranes. In this area, we focus on stimuli responsive nanosystems, nanosystems that can cross the blood-brain barrier for neurotherapeutic applications, the blood-ocular barrier for the blood-retinal barrier, sorry, for intraocular drug delivery to cure blindnesses and diseases that cause infections in the posterior segment of the eye. We look at intravaginal systems, getting drugs through the vaginal tissue, as well as transdermal, topical drug delivery. So at the tissue level, we have projects that focus in these areas where we're getting nanostructures across the biomembranes. Then we moved on to more complex level, which is a cellular level. In here, we looked at chemotherapy. When I speak about chemotherapy, it's not chemotherapy only in the sense of oncology application. It's the chemotherapy of TB, malaria, HIV. These are all kinds of, where we have multiple drugs that need to be dosed at once, to cure or disease or illness. And here we looked at more of, the targeting ability. How do we target the cells that are responsible for causing disease and illness? Chemotherapy, looking at tumor microenvironment targeting, tumor cell penetration, as well as, targeting using the body's immune system and host-directed therapies for tuberculosis, malaria, HIV, tropical diseases also now recently included, as well as the development of theragnostics. Because most times when you're developing these targeted systems, there's nothing stops you from putting a diagnostic molecule within these systems, that you can have a joint therapeutic with diagnostic capability in the system. That's at the cellular level. And then we moved on then also to the tissue or the extracellular matrix level. And here we're talking about neurotrauma, rewiring the neural circuitry, nanocues for neuro or spinal cord injury and peripheral nerve injury. Professor Kumar, who's joined on this webinar, will give more detail on these aspects of neuroregeneration. So as we see where we're starting off, the complexity increases as we go along in terms of nanomedicine. And at the ECM level, we're also looking at, how do we regenerate neural tissue? We also look at now liver and other organs that can be regeneratable, and also using 3D bioinks. That brings on to the next level, which is 3D bioprinting, or the additive manufacturing spin is getting on quite a bit. That's the next revolution. Where nanotech started, I think 3D printing, bioprinting is going to take over. So how do we combine nanotechnology or nanomedicine with 3D bioprinting, the classical 3D printing, where we have a biological component to it? And here we're looking at wound healing, tissue engineering, scaffolds, and devices. And we're working also closely, building partnerships with our colleagues at the University of Mauritius and Archana and team in the wound healing area and wound healing space as well. How do we get these 3D bioprint, the next wound healing, pseudo skin, biodegradable systems where we can have advanced wound healing? And the wound healing also ties in a lot with the neurotrauma application, as well as diabetic foot ulcers, as Archana has mentioned, etc. And then the next level or the next trajectory is obviously the genetic level. And here, I mean, the classical example that we've seen where nanomedicine has come to the fore in the genetic level is with the pandemic. You know, the mRNA vaccines that you've seen and being delivered using nanostructures. This is where the world is playing at the current moment, you know, delivering different genetic materials to see how we can get gene therapy going. And obviously for those kinds of therapies, we need nanostructures to get to the nucleus of the cells. So in a nutshell, just gives you an overview of where we are, you know, in terms of our research in nanomedicine. We have not left any of the levels that I've described earlier on. We are involved in all of these levels, but just to show you that the complexity of the field is getting more and more complex as we go along. And as an African continent, as a continent that wants to drive on these areas, we need to make sure that we keep in line with the regulatory environment, the commercial needs, as well as the scientific needs that are happening at a very fast pace. So that's just a snapshot of the research, and I think Prof. Tutoy will also cover some of the areas of technical areas of research that we do at the WADAP. Most of our research is obviously linked to ex vivo and in vivo preclinical testing models. Again, Prof. Kumar will share more on you on this. I'm just going to give you a very graphical overview for now, where we look at different animal models at the in vivo level preclinical testing. Important I share this with you is that, you know, lots of promises have been made in the nanomedicine space, but the question still begs is why have we not seen any of these into the market space? And the answer, one part of the answer to that is that, you know, the biological interaction of nanostructures with the biological systems, the fundamentals are still not yet ironed out truly. And that's why we have these models and in vivo preclinical testing in very detailed way to actually, allay the fears of whether the safety and toxicity of nanoparticles. I mean, if you look at carbon nanotubes, they have been around for ages, but yet there's still questions on the controversy of whether carbon nanotubes are safe or not. So, you know, in other fields, nano has moved way ahead because it's a system, or you'll be using materials or applications that are static. In the biological space or nanomedicine space, it's a dynamic area where the biological system itself is changing on a daily basis. I mean, you look at antibiotic resistance and how these biological and bacteria are becoming resistant to our antibiotics that was developed years back. And still today, we're looking for solutions for that. So when we're hitting a moving train in terms of a biological system, we can't work with static nanostructures. And these nanostructures need to be aligned to the biological systems that are moving at a very fast pace. And that's why you tend to find sometimes, you know, as scientists, you tend to overpromise on certain what nanotechnology can do for the market. And yet we don't see on the out end, you know, the actual commercial impact that's been made. And it's not that we don't have the answers. It's just that we, you know, not rushing certain things to get to the bottom of the biological systems and tying it up with the actual nanostructures that we develop. We have over 300 international accredited publications in the space of drug delivery, tissue engineering, biomaterials, and nanomedicine. 40% of these publications are in the area of nanotech, and as a group, we continuously to publish in these areas. Some of the research that I've given you as a graphical abstract here today is available in the literature for you to read, and we welcome and we're happy to share any of this with any of the audience and anybody wanting to read further. Prof. Kumar and Prof. Tutoy will give you some more detail on some of the exciting technologies we're currently working on. Just to say that in addition to the ISI accredited publications that we have in this area, we also have patents that we filed to tick the box of the commercial angle that we look for. You know, we have 43 patents filed, 23 are awarded in international jurisdictions, which are the USA, Europe, Asia, India, China, Japan, and even in ARIPO, the African continent. 20 are in progress under examination. 30% of this is in nanotech, I will say nanomedicine. And obviously, the questions that whether these will reach the market or not is the next big question. And that's where I take the Conde's comment about, it's a time for action. So we have these patents on the continent. I mean, we are certainly one group, or there's many others that also have this kind of patenting activity going on. But why is it not translating into where we need it to be? And one of the key components, the gaps that we do see is actually on the training and capacity development. How do we build capacity so that you have 3 or 4 scientists working on these technologies, but then we need people to then run with that in commercially, in making sure that they actually reach where they need to be in terms of the next generation of nanoscientists or nanopharmaceutical scientists. So in conclusion, I just want to say the R&D efforts continue to contribute to innovation, and innovative products contribute to economic growth. So we are at the area of good research and innovation, but what we have not realized in a big scale as yet, and what we need to do is the innovative products area to realize the economic growth. And it's a vicious cycle or a good cycle in that when you get products, you get economic growth. Economic growth results in more money gets more investment and more into research. We need to expand our innovation capacity and human capital development. And we see this a lot at WADEP. We have a lots of students from the African continent that join our team annually, from all countries, I think 9 different countries on Africa. And the sad part is that we develop this high potential people, and then the market cannot absorb them. And that way we need to work together in partnership with industries, the funding models on how we fund research in nanotech. Do we look at large research centers as we've done in the past? Is that how we're making impact? Or rather, should we look at funding principal investigators with good projects so that we could get more focused action, more focused research going out, and then developing the large-scale commercial interest later on, and flowing more money into human capacity development, because that's what we need for the future of nanomedicine to really be driven at the patient's bedside. More specific targeting, greater safety and faster development is required. When we talk about safety, we also need to bring into account our regulatory environment. We cannot work in isolation of the regulatory environment. Regulatory pathways are critical for nanomedicines. Where is our regulatory bodies in terms of absorbing nanomedicine technologies and approving these for patient use? And that needs to move. I mean, the science is running away and the regulatory is remaining behind, and that we need to tie them all of that up. To focus on bioresponsive or self-regulated systems is also important, and intracellular gene and bioactive delivery. The world is moving to genetic material and gene therapies, and we need to make sure that we don't miss that boat. And therefore, we have at the WADEP at least, we have a program where we're looking at delivering genetic material in a much more advanced way. And these are available on the African continent so that we are ready for when we get the other pieces of the puzzle together. And not to avoid or to forget that electronics will also play an increasing role in biomolecular delivery, electronics and 3D bioprinting as a way forward. So colleagues and attendees, I mean, this is what I have to share with you this morning. I'm looking forward to some of the discussions we had earlier that raised up to this webinar of, online master's degrees, Pan-African nanotechnology, online masters in nanomedicine, to help build that nanomedicine capacity on the continent. Thank you for listening to me, and I look forward to the rest of the seminar this afternoon and this morning. Thank you.
Thank you very much, Aya, and thank you for ending with the master nanotechnology that we want to build up. And for this will come up in the discussion session, because UNECA has been leading this. And thank you also for pointing out the dynamic aspect of nanomedicine. And this is very crucial, and training and getting decent jobs for our human capacity that we are building is also very important. Now it is with great pleasure that I introduce to you Dr. Itisha Chaman. She's the youngest one in the team today, that is for the webinar today. And this is also because we promote the SDGs, and this forms part of what we do, that is quality education. And Itisha, she did her master's MNH degree at Queen Mary University, then joined us for a PhD at CBBR and stayed for after a PhD for a postdoc. And I would like to say that she just won the UK Alumni Award 2021 for social impact for the work that she's doing. And I'm very happy because she participated in social impact category for a specific reason, because we want to show that whatever we have been doing has got potential social impact. That is, the community can benefit from that. And Itisha, it's Your turn now. You are on mute. We can't hear you.
Okay, so hello everyone, I am Dr. Itisha Chuman, and as Dr. Balakstino said, yes, I have been a PhD student at the center, and I have recently joined the center as a postdoctoral research assistant. And today I'm going to talk about hydrogels in tissue engineering, so the synthesis, the development of hydrogels in TE. So as the word says it all, hydro means water. So these are gels that contains over 90% of water. They are made up of long chains of polymer, which are cross-linked by either physical or chemical cross-linkings. Now, a few examples of these chemical cross-links are radical polymerization in the presence of carbon-carbon double bonds, or shift-base cross-linking in the presence of a carbonyl aldehyde or amine groups, and disulfide bonds in the presence of thiol groups, or physical crosslinks such as hydrogen bonding, ionic interactions in the presence of carboxyl or amine group, or in the presence of ionic groups, and as well as hydrophobic interactions. Now, these long chains of polymer can either be synthetic, semi-synthetic, or natural polymers. However, a lot of research is driven towards natural polymer-based hydrogels, owing to their biodegradability, low immunogenic, and as well as low cytotoxic properties. Now, to this effect, we have a wide variety of polysaccharides and polypeptides, which have been used to synthesize these hydrogels. As for example, hyaluronic acid, chitosan, arginate. They provide functional groups such as carboxyl groups, amine groups, for cross-linking. Or we have cellulose, dextran, which are naturally available. They contain large amount of hydroxyl groups, which can be modified to allow cross-linking. Or silk fibroin, which is a polypeptide with self-assembling properties. Now, hydrogels have been used in the biomedical field for a wide panoply of applications, such as contact lenses, drug and gene delivery, wound dressing, biodevices and biosensors, as well as regenerative medicine. In regenerative medicine, that is tissue engineering, hydrogels act as scaffolds, essentially to provide structural integrity and bulk for cellular organization and morphogenic guidance for different tissue engineering applications such as skin, bone, or even cartilage regeneration. They mimic the 3D structure and the physicochemical characteristics of natural extracellular matrix. So let's look at the design criteria of hydrogels in TE. Hydrogels have the ability to provide both a 2D and 3D platform for cell culture. However, 3D outweighs 2D since it better predicts in vivo cell behavior. Now, 2D, they confine cells to a more planar and spread morphology with restricted interactions with cells, surrounding cells, and matrix. And 2D, static 2D cell culture, they favor uniform diffusion of nutrient and growth factors. However, a diffusion gradient is favored in 3D cell culture. Now, 3D hydrogels can also be used for delivery as delivery vehicles for cells, growth factors, and therapeutic drugs. Now, the physicochemical properties of hydrogels, they play an important role in cellular behavior. To start with, pore size, porosity, pore interconnectivity. Different techniques are used to render these hydrogels porous, as for example, solvent casting, particle leaching, gas forming, freeze drying. And freeze drying is mostly used to to improve pore interconnectivity, which is important for cell culture. This is an example of a cell grown onto chitosan hydrogels, porous chitosan hydrogels. So in this example, the size and porosity of the chitosan hydrogels have shown to influence cellular infiltration and viability of human fibroblasts. An increase in the pore size from 32 to 92 increased cellular infiltration, and high porosity also improves cell viability so that nutrients can pass through and reach the inner cells within this structure. Other studies also show bigger pores improve rate of chondrocyte proliferation in gelatin hydrogels and improve GAG synthesis and collagen synthesis. So it shows that the porosity plays a major influence the cellular behavior in a hydrogel system, which is very important to cater when developing these types of hydrogels. Now, other properties of the hydrogels are mechanical stiffness and surface properties. It is known that cells on a matrix, they attach via integrin containing focal adhesion link to their cytoskeleton. So it pulls on the matrix and it sends the resistance, and this increases the tension in its structure. So it tends to spread out on the surface. So this is an example of a human fibroblast seated on a 2D structure, a hydrogel scaffold. So obviously you can see while increasing the compressive modulus, the spreading increases and also the proliferation increases. The same applies to RGD density, that is the surface properties is modified by either, you can either chemically immobilize these RGD sequence. RGD sequence are adhesive ligand, which will help adhesion of the cells. So as you increase the density, the cell spreading increases. So it can be either chemically immobilized or physically encapsulated. However, you can see this is a 3D platform, so chondrocyte seated on a 3D hydrogel. The chondrocyte was seen to spread, on the contrary, on a lower compressive modulus. So the mechanical properties of hydrogels is very important to modulate them. And how do we do that? Is either by varying the cross-linking density, by either increasing or decreasing the cross-link, the amount of cross-linker, or modulating its substitution of functional groups, or even the polymer concentration while synthesizing these hydrogels. Now we're going to talk about more on the smart hydrogels that we have in tissue engineering. So first, stimuli responsive hydrogels, which is quite an interesting hydrogel system. They show distinct changes in the properties with external stimuli like temperature, pH, solvents, ions, redox, etc. They can be used for in situ hydrogels under physiological conditions or for release of therapeutic drugs, proteins, etc. Now, this is an example of a pH redox sensitive hydrogel based on BSA. So BSA in the native form, they have disulfide groups. So these are interesting groups that can be used or introduced in other hydrogel systems so that you can induce the pH or redox responsiveness of the hydrogels. So it has disulfide groups in its native form, which upon reduction can form thiol groups and then can reform the disulfide bridges upon oxidation. So this will have an influence on cross-linking at different pH. So between pH 4.8 and 8, it cross-link, and it does influence the swelling properties of the hydrogels. Now, another commonly used thermoresponsive hydrogels is based on polynipan. So the thermoresponsiveness of polynipan is attributed to its hydrophilic amide group and hydrophobic isopropyl group, which above its lower critical solution temperature forms intermolecular hydrogen bonds or hydrophobic interactions so that they gel at physiological temperature. So these are interesting systems that can be used in TE to for example, form in situ hydrogel so that when injected, it can instantly form the hydrogel under physiological condition. So this brings us to another type of hydrogels, which are injectable and self-healing hydrogels. So one of the property of injectable hydrogels can be in situ gels, so that the gel precursor is injected at the wound site and it cross-linked under physiological condition. Now, the injectability of these gel precursor or hydrogel itself depends on their shear thinning and self-healing properties, so that the bond breaks during injection and reform after injection, which necessitates dynamic covalent bonding, that is physical interactions. Now, one common example is shift-based reactions, which is reversible. Hydrogels, they require controlled gelation kinetics, that is, they gel within a given time frame. They should gel within a given time frame. Another system which proves to be effective for different cross-linking mechanism is a double barrel syringe, which contains solution A and solution B separately. And when mixed together, they form the gel you can use a mixing tip so that it instantly mixes the gels into the wound, and in the wound it gels instantly. So the gelling kinetics is also crucial in such a system. This is an example of a hydrogel system that we are currently working in our lab. So it is a injectable hydrogels using a double barrel syringe, and also it has self-healing properties. So once the bond breaks, it can reform. Now, these types of hydrogels are very interesting for deep wounds, deep open wounds, and also for cartilage tissue regeneration. Now, another emerging type of hydrogels is peptide-based hydrogels. The unique properties, the gelation mechanism by self-assembling. This is an example of a peptide primary sequence. This is just one example. There are a million others. So it has a hydrophilic and hydrophobic domain along its amino structure, which can rearrange into what we call beta sheets via hydrogen and hydrophobic interactions. But we also have other peptide secondary structures, such as beta hairpin, alpha helix, or coil. Now, the general mechanism for this hydrogel synthesis is a concentration-dependent nucleation followed by fiber elongation, so that forms long fibrils, and then a lateral aggregation to form these mature fibers. So these are interesting hydrogels, which we can investigate further, which are being investigated in our lab as well. And last but not the least, hybrid hydrogels. So they provide the ability to tailor properties of hydrogels, which opens up to a wide range of possibilities and applications in TB. So it can have synergistic properties together. So hydrogels can be loaded with either nano microspheres or even nanoparticles, which I forgot to mention. So these allow for controlled delivery of therapeutic drugs or bioactive molecules, and also by incorporating the molecules into the micelles or liposomes, it increased the loading efficiency of hydrophobic molecules. So it increased the loading efficiency within the hydrophilic structure, hydrogel structure. Various techniques have also been used to incorporate nanofibers and microfibers. Obviously, we work with nanofiber, we do electrospinning in the lab. So we are trying to, we are looking into hybrid scaffolds where you have the hydrogels incorporated with these nanofibers, either to improve the mechanical properties or to induce synergistic biological properties of an electrospin mat or a hydrogel. So you have sandwich in a sandwich model, either it is dispersing the hydrogel, or in terms of electrospin mats with each fiber are actually hydrogels, which can retain water. Other type of hybrid hydrogels are polymer protein composite hydrogels. So they have been synthesized by either you conjugate the protein on the backbone of the polymer, or by polymerization with two solutions of polymer in the proteins. The presence of proteins, it induces self-assembling properties to the hydrogel system. Also, hydrogels have been incorporated with reinforcing materials, especially for bone tissue regeneration, so that you improve the mechanical properties. So hydroxyapatite, ceramic, clay, and cement have been added to these hydrogels. So to conclude, the synthesis, characterization, biological testing of hydrogels for tea applications, it obviously requires a interdisciplinary approach, as you can see, polymer chemistry, design of hydrogels, surface chemistry, cell biology, imaging techniques, and so on. But to date, only a few hydrogel systems has reached commercialization due to the reproducibility of the system on a large scale. So a trade-off is required between complexity of the system and reproducibility. So however, it is important to point out that the chemistry behind hydrogel synthesis do not require high technologies. So it is important for Africa to investigate into such systems, given the large panoply of natural materials that we have, which can help us to build up on and develop a wide variety of hydrogel systems at affordable cost, designed to meet requirements for a wide range of tissue applications. So on this note, I will leave you to reflect, and I would like to thank you for your attention, and will be happy to answer the questions at the end of the session. Thank you very much for your attention.
Thank you, Itisha, for this. Our next speaker is Dr. Noshin Gunu. Dr. Noshin is also a, if I can put it that way, product of the center. She received the L'Oreal Rising Talent Award last year, and she is right now working as the L'Oreal Research Fellow at the center. And she's going to give to you an overview how we can use in vitro techniques as a predictor for in vivo outcome, which is very important for the African continent because it allows us to reduce costs. Noshin, up to you.
Thank you. Thank you for the kind introduction. Itisha, can you please unshare your screen? Thank you. Good morning, everyone. So now that you have been introduced to the concept of tissue engineering and scaffold design, I will carry on with in vitro techniques and how these can be used to predict the performance of scaffolds in vivo. But first of all, let us differentiate between the terms in vitro and in vivo. In vitro refers to all cell-based techniques, whereas in vivo refers to animal-based studies. Now, for any product to reach a commercial market, there are various stages which are involved. The first stage or step involves the development and the design stage, which is followed by in vitro screening, in vivo screening, and clinical trials. Now, all these stages or steps, they occur in a sequential manner, and it is very important not to bypass any of those steps. Now, you will know that as we go along each of those steps, the time and money associated with each of those steps also increase. Now, for a particular biomedical product, it may that when it undergoes in vitro testing, it may, for example, not satisfy all the criteria. And at this stage, we will say that the biomaterial or biomedical product has undergone an early stage failure. Now, if this happens at this point of in vitro screening, the product has to go back to the development and design stage and go for the whole process again. Now, why is it important to go for in vitro tests before going for in vivo screening or in vivo studies? There are two main reasons. The first reason is because of ethical considerations. Now, according to the Russell-Lindbergh principle of the 3 R's, where one of the R's stands for replace, it is crucial to replace all in vivo studies by in vitro studies as far as possible. The second reason is because of cost implications. In vivo studies cost a lot more than in vitro studies. Now, just to give you a little bit of background on the tissue engineering strategy, now tissue engineering, the first step of tissue engineering consists of isolating cells from a patient's body, expanding the cells in vitro using growth factors or scaffolds to finally form the tissue, and then the tissue is implanted into the patient. Now, for any tissue engineering application to be successful, the scaffold is very important. The scaffold plays a crucial role in determining the success of any tissue engineering application. For any tissue engineering application, scaffolds need to possess a number of properties or criteria. For instance, the scaffold should be biodegradable. It should be porous. It should have appropriate pore sizes. It should also have good surface properties and topography so as to promote cellular response. It should also have good mechanical properties so as to be able to withstand biological forces and maintain physical integrity during the whole tissue regeneration process. And also the scaffold should be biocompatible. Now, what do we mean by biocompatible? It means that when the scaffold is implanted into the patient or in the host system, it should not cause any toxic reaction or any illicit reaction. Now, there are many in vitro tests that we can use to test for the toxicity or biocompatibility of a scaffold. One of the most conventional and most often used method is the live and dead staining method. Now, let's say you want to test the toxicity or biocompatibility of your scaffold with respect to fibroblast cells. The first step will be to grow fibroblast cells in a flask. Once your cells have reached confluency, you can prepare your scaffolds in your well plate. So first you need to cut your scaffolds into small pieces and place them at the bottom of your well plate. Now, it is very important at this stage to leave some of the wells empty. Those empty wells are going to serve as control. Once you have prepared your well plate, you can then seed a known number of fibroblast cells into the wells, and then you can incubate for a given period of time, let's say for 3 days. After 3 days, you're going to add the live and dead staining solution, and you're going to incubate for 10 minutes. Now, the live and dead staining solution consists of 2 different dyes. So you have fluorescein diacetate, FDA, which is going to stain the live cells. And you will also have propydium iodide, PI, which will stain the dead cells. So after 10 minutes, you're going to view the cells under the fluorescence microscope. And you can expect to see an image like this, where basically you have red and green dots. Now, the red dots correspond to dead cells, and the green dots correspond to live cells or viable cells. What you can do at this stage then is to count the number of live and dead cells. And you can use this equation here to calculate the percentage cell viability. Now, if you recall, you started with 2 groups where you had a control group and a scaffold group. Now, if you, at the end of the calculation, you reach with a similar percentage cell viability for your control group and your scaffold group, this would indicate that your scaffold is non-toxic. On the other hand, if you have a significant reduction in cell viability for your scaffold group compared to your control group, then this would indicate that your scaffold is being toxic to fibroblast cells. Now, what is the principle behind live and dead staining? So if you recall, we used fluorescein diacetate to stain the viable cells. So this is the structure of fluorescein diacetate. Fluorescein diacetate is going to enter the cell membrane of cells, and in the presence of esterases, this fluorescein diacetate is converted into fluorescein, and this accounts for the green color. Propydium iodide, on the other hand, is not cell membrane permeable, which means that it cannot enter the membrane of viable cells. Therefore, it will stain only dead cells. So this propydium iodide dye is going to enter the cell membrane of dead cells, and it's going to intercalate with the DNA. And this is going to give rise to the red color. Now, apart from all those properties that we have mentioned before, scaffolds also need to have some additional properties. For instance, it should not cause any significant inflammatory response when it is implanted in vivo. In other words, we should not observe any significant swelling or redness when we implant the scaffolds in vivo. In addition, the scaffold should promote the formation of blood vessels for nutrient transport and removal of waste products. Therefore, the scaffold should promote angiogenesis. And one more property is that the scaffold should promote cell migration, especially for skin tissue engineering and to fill the gaps in bone defects. Now, how do we test for the inflammatory response in vitro? Macrophages are a very important cell in our immune system. And depending on their functions, they can be categorized into 2 different subtypes. So we can have the pro-inflammatory phenotype, M1, and we can have the second type, which is the anti-inflammatory phenotype, or also known as the pro-healing phenotype, M2. Now, we can differentiate between these two phenotypes based on their morphology. So if we look at the scanning electron microscope image here shown on the right-hand side, you will see that image A shows unstimulated macrophages, which are here denoted as M0. So if you look at the structure, you will see that M0 macrophages, they are rather small and they are rounded in shape and they lack cytoplasmic extensions. On the other hand, if we look at M1 macrophages, which are shown in image B, these M1 macrophages, they adopt an amoeboid shape and they have cytoplasmic extensions. If we look at image C and D, these are for M2 macrophages, and we see that M2 macrophages can have two distinct morphologies. They can either be in the form of an elongated structure, as we can see in image C, or they can adopt a multinucleated giant cell structure with cytoplasmic extension, as we can see in image D. Now, apart from morphological differences, we can also differentiate between these two phenotypes based on the cytokines that they produce. For example, the M1 macrophages are going to produce the pro-inflammatory cytokines of the type TNF-alpha, IL-6, IL-1, and so on. Anti-inflammatory phenotype macrophages are going to produce pro-healing or anti-inflammatory cytokines like IL-10, TGF-beta, VEGF, and so on. Therefore, depending on the cytokines that they produce, we can characterize the type of macrophages which are present. And in vitro, we can quantify the amount of cytokines which are produced using commercial ELISA test kits. So the first few steps will be the same. So first, we're going to grow macrophages in a tissue culture flask. We are then going to prepare our scaffolds, leaving some of the wells empty. And then we're going to place the macrophages or seed the macrophages into those wells. We are then going to use the supernatant and we're going to carry out ELISA tests to quantify the cytokines which are produced. Now we can, for example, determine the concentration of cytokines which are produced for different scaffolds, or we can determine the concentration of cytokines for a given scaffold, but over different time intervals, like we have, shown here in the graph. Now, as we have seen before, angiogenesis is also a very important property for scaffolds. One of the most used assays to test for the angiogenic potential of scaffolds is the tube formation assay. So how do we go about this test? Basically, we're going to seed endothelial cells onto the scaffold, and if the scaffold has angiogenic potential, it will cause the cells to rearrange and to form tubes and nodes. So after some time, we're going to view the cells under the microscope, and on the right-hand side, you can see fluorescence microscope images showing networks which are formed. So these networks are actually the tubes, and the intersecting points are actually the nodes. And you can quantify, you can count the number of tubes and nodes which are formed for the different scaffold groups. So if you look at the images here, A, B, and C, and also the graphs, we can conclude and say that scaffold C has a much higher angiogenic potential in comparison to scaffold B or A. Now, in skin tissue engineering, cell migration is also a very important aspect which we need to consider. Now, if you look at this figure here, it shows a wound where on either side you have cells growing, and in the middle you have an area which is empty and does not contain any cell. So this is basically the wound. So the void area is representing here the wound. So for the wound to heal or to close, what needs to happen is that cells on either side of the wound need to migrate towards the center of the wound and close the wound. So in vitro, how are we going to mimic the wound? So basically, we're going to grow first a confluent layer of either keratinocytes or fibroblasts in a well plate. Once the cells have formed a confluent layer, we're going to use a pipette tip and we're going to make a scratch in the middle of the well to form a wound. And then we're going to monitor the migration of cells into the void area over different time intervals. So here we have optical microscope images showing how the wound or void area is progressing over time. So at T0, you can clearly see the void area, and as time goes by, you can see that the void area is filling up with cells. And we can calculate the percentage wound closure using the following equation. Now, So far, all the conventional tests that we have looked at have used 2D cell culture models. But as we have seen in the previous presentation, 3D cell culture systems better mimic the native microenvironment. Therefore, recent developments have led to novel innovative approaches to cell culture techniques. And one of the most innovative approaches is using 3D spheroids. So what are 3D spheroids? 3D spheroids are basically cellular aggregates whereby we have dense cell-cell and cell ECM interactions. So a spheroid, it can consist of several thousand of cells, as you can see in this representation here. Now, we need to be very careful whenever we are designing or developing spheroids. The overall size of spheroids should not exceed more than a few hundred micrometers, because otherwise the center of the spheroid is going to be necrotic. Now, these spheroid systems have shown improved biological properties in terms of having a more stable morphology, lack of polarity, increased proliferative activity, and physiological metabolic function. This figure here shows an optical photo of spheroids where you have the average size of the spheroids is approximately 343 microns, and each of those spheroids contain approximately 2,000 cells. And here you have a scanning electron microscope image where you can see the spheroid cell migrating onto the nanofibrous mat. Now we can also use a 3D cell culture system to test for the angiogenic potential of scaffolds. We can use, for example, the sprouting assay. So in the sprouting assay, we're going to fabricate endothelial cell spheroids. These spheroids will then be seeded onto scaffolds, and then we're going to monitor the migration of cells from the center of the spheroids towards the nanofibrous mat or your scaffold. And this here is known as the sprout. So what we can do is we can quantify the length of the sprout over time. Here we have a colored image showing the sprouts and the area occupied by the spheroid. So in green here is the initial spheroid area, and in red you can see the area which is occupied by the sprouts, and in blue you have the sprout length. So you can quantify, for example, the area occupied by the sprouts and the length and number of sprouts, and this should give you an indication about the angiogenic potential of the scaffolds. And we can also use spheroids to have an indication about the cell migration of scaffolds. We can use, for example, fibroblast spheroids, and we can seed the fibroblast spheroids onto scaffolds, and we can monitor the migration of cells onto the scaffolds. And we can plot, for example, a graph of the migration distance with time. And this can give you an indication about how much the scaffold is promoting cell migration. So conventional in vitro tests have so far involved the use of 2D cell culture, followed by animal models, and finally clinical trials in patients. This is here represented by the longer red pathway. The use of 3D cell culture systems, which is here represented by the green and therefore shorter pathway, allows scientists to bypass the lower efficacy 2D models, minimize the use of large-scale and expensive animal studies, and improve clinical research by identifying promising scaffolds more rapidly. Therefore, 3D cell culture systems are more affordable for the African continent compared to the use of large-scale animal models. With this, I would like to thank you for your kind attention.
Thank you, Noshin. Yaya, I will let you continue the session.
Thank you, Archana. Thank you for that insightful presentation, Nishin. I think next on the agenda we have a presentation now by Prof. Lisa Dutoy from the Virtual Advanced Drug Delivery Platform. Lisa works close with our team in this area. She's going to share some information on the tissue engineering aspects. And Lisa, welcome, and the platform is yours.
Good morning, everyone. I'll just share my screen for a moment and then I'll open my presentation. So today I'll be discussing experimental techniques for tissue engineering and regenerative medicine. Thank you to Prof. Chinara for the introduction earlier on as well as my team members and all the students working on these projects. So I'll just go into a bit of a definition. We've already discussed tissue engineering. You all know what it's about. Introduced many years ago as an interdisciplinary field which applies principles of engineering and life sciences towards development of biological substitutes, restoring, maintaining, and improving tissue function. So, of course, tissue engineering aims to induce tissue-specific regeneration processes, and it evolved from the field of biomaterials development. So what we're doing is combining scaffold cells and biologically active molecules into functional tissues. Now, just to clarify in terms of what regenerative medicine is, that's a broad field that also includes tissue engineering, but also incorporates research on self-healing, where the body basically uses its own systems with the aid of foreign biological material to recreate cells and rebuild tissues and organs. Essentially today, these terms are quite interchangeable, but what we must focus on is for cures rather than treatments for complex and chronic diseases. So as scientists, of course, we really do want to understand the underlying mechanism behind tissue engineering and regeneration. So groups of cells obviously generate their support structure, which is the extracellular matrix, which is a scaffold. And the scaffold is what is supporting cells and relaying messages from various signal molecules. And so cells receive messages from sources in their local environment, and this signal initiates a chain of responses to determine the cell's fate. So understanding the mechanism of the individual cell's response to the signals and interaction with the environment, we as researchers are manipulating these processes to now mend damaged tissues or create new ones. So as you all know, and as we've discussed, the process is beginning with building a scaffold from a wide set of possible sources, such as your polymers and proteins. And then cells and or growth factors can be introduced. And you need to create that correct environment to enable tissue development. So you can create this in one step by a process of cell assembly. Another approach is to also use an existing scaffold. So here I'm just going to go through the approach we use in our research unit for tissue engineering. And this is using a bone scaffold as an example. So what you want to do is first select and manufacture your biomaterial for using for the tissue engineering approach. And following that, you're going to look at various fabrication approaches for designing a nanostructured scaffold for the tissue engineering. You can be using electrospinning, phase separation, sol gel approaches, SLS, 3D printing, just as examples. And then what's important after that is also to discern which biorelevant nano-sized additive you'll need to include to promote the wound healing process. And very important after that is the actual testing in a defect site. And this is where the in vivo testing models also come into play, which Prof. Kumar will be discussing further as well. So, I mean, in the past, 3D printing was something futuristic, and obviously now it's moved into the 21st century where it's becoming very important in the pharmaceutical sciences for creating personalized medicine. It's obviously a convenient and becoming cost-effective manufacturing approach for personalizing pharmaceutical, medical, and dental products. Now, if we look at 3D bioplotting specifically, which Prof. Chinara did introduce already, that's a specialized arm of 3D printing that's employing biological materials in computer-aided tissue engineering to create structures for enhanced biomimicry. So the goal is to get an effective combination of cell and bioactive components for restoration or improvement of your biological functionality. So last year, we were responsible for editing a book which we published on the convergence basically of your 3D printing and nanotechnology. So we discussed the important applications of your 3D bioplotting in pharmaceutical research. Where nanotechnology comes into this is the design and fabrication of scaffolds at the nanoscale for actually controlling the spatiotemporal release of your biological factors, because it's creating that environment that resembles a natural extracellular matrix. The convergence of your biofabrication by 3D bioplotting with nanotechnology is a largely unexplored but growing focus for constructing directional customizable biomaterial arrangements to promote your tissue regeneration, as well as having the potential for controlling bioactive delivery in the region where your scaffold is implanted. So this impetus for your nanoprogression in 3D printing is because your nanostructures, and these could be your nanoparticles, nanotubes, and nanofibers being smaller than 500 nanometers, they can stimulate your native biological systems, enabling tissue regeneration via cell growth. So your biopolymer matrix and your nanostructure interactions allow for a nanostructured material that has enhanced functionality, and very importantly, the mechanical properties which are largely introduced by your nanostructured components. So this just outlines the approach we use at Product for our 3D bioplotting. Firstly, and maybe most importantly, is actually defining and designing, formulating, conceptualizing the bioink, which is comprised of your biomaterial matrix, the nanostructures, which you would include, which can be natural or synthetic in terms of your biomaterial, and then biological components. This is input into a computer-aided design together with your 3D printing software or bioprinting in a layer-by-layer approach. So what I'm going to go through now is a few of the projects that we have done in our labs, where it's actually integrating your 3D bioplotting and tissue engineering techniques, as well as the nano enhancement approach that has been employed in that instance. The first example here is a 3D bioplotted PVA PAA hydrogel that's a scaffold for the delivery of indomethacin, which is an anti-inflammatory. So the addition of your actual anti-inflammatory agent to your 3D bioplotted scaffold allows for the acute treatment of site-specific inflammation, as well as then providing structural support long-term to allow for tissue repair and rehabilitation. So what was actually designed is this scaffold, which was then nanostructured via semi-interpenetrating network composed of your PVA and your PAA. So your nanostructured IPN allowed for the production of a dense hydrogel matrix that had strong mechanical properties and efficient drug loading. So here you can see some microscopic images of the actual matrices developed, and those were subjected to testing on biotested to determine the physico-mechanical properties, matching those with what is required inside the body. And we also saw that with hydration of the scaffold, there was inflammation-sensitive hydration with corresponding release of anti-inflammatory agents. Another example which we looked at is here a bio-inspired 3D bioplotted device for treating burn wounds. Now, in Africa, burn wounds are a very major problem and can, besides resulting in great debilitation, time off work as well, which is a great economic problem. So what was proposed here is a 3D bioplotted wound healing device that has superior wound regenerating properties, and it incorporates cell seeding and growth factors as well. Essentially, they use 3 layers, and it provided controlled release of your active compounds. The nano enhancement was the various nano and micro architectures incorporated via your cell seeding and growth factors as well, your biomimetic polymers, active compounds, and biomolecules. So just showing the fabrication approach for your 3D bioplotted device. Firstly, an initial hydrogel was created. This precursor hydrogel was partially cross-linked. So this was the first cross-link between lysine and alanine together with your 4-arm peg to create the initial hydrogel, which was then 3D bioplotted. And this was then secondly cross-linked to obtain a fully cross-linked scaffold via UV light. Another approach which we looked at as well was a 3D bioplotted implantable drug delivery scaffold for application in bone tissue engineering. So, of course, traditional treatments for your bone defects include your orthopedic implants, allografts, and autografts, but here you have a lot of problems with infection, poor healing following invasive surgeries, not enough donors, as well as morbidity at the donor site. So what was proposed here was a 3D bioplotted pseudo-bone drug delivery system, which was then inserted at the site of the fracture. What was actually used was the clavicle model. The system composed of a copolymeric biomatrix, and it was also based on a thermoresponsive gel, which was loaded with a statin drug, which basically stimulates bone regeneration. So what was firstly undertaken was that a 3D model was constructed, and this was then biofunctionalized with various osteoinductive signals, growth factors, and peptides. And this was then implanted at the site of the fracture. Ex vivo evaluation was undertaken in a bone model, and various techniques were employed to analyze the success of this approach via microscopy, ultrasound, and X-ray. So you can see from the SEM images, you have your nanoarchitecture and your porous nature that's created by your scaffold, enabling your nutrient diffusion, cell attachment, and proliferation. X-ray and ultrasound images were done on the human clavicle to show the actual healing from before the fracture to after the fracture, and following the successful 3D scaffold treatment. SEM also showed the osteoblast-like cell attachment to your actual bioplotted scaffold. The next system which we're looking at is fluid absorptive alginate chitosan bioplatform as a potential wound dressing application. So this was developed because of the need to actually improve your performance properties of wound dressings to obtain more enhanced wound healing. And what was done here was a specific bioplatform approach using partial cross-linking, free-drying polymerization, as well as functionalization. The nano enhancement here was the in situ formation of your partially cross-linked interpolymer complex hydrogel using chitosan alginate, nano to microparticle sizes, as well as the use of an adhesion motive for development of a self-assembled scaffold. The bioplatforms developed showed optimal fluid absorption, porosity, degradation, and mechanical properties, and also had desirable drug delivery properties, cytocompatibility, and wound healing performance. So this just shows the approach for fabrication of your bioplatforms. The combination of your natural polymers, your alginate and your chitosan, with your adhesion motive to create your self-assembled model. And you can see the morphological change in your scaffold with the self-assembly of the polymers together with the adhesion motive. The increased porosity is very important, like we said, for mimicking that extracellular matrix for promoting tissue regenerative capacity. Importantly, we looked at the performance of these bioplatforms. Here we actually created wounds within the rat model, which were tested with the platform, and it showed improved properties in terms of its degradation, fluid uptake, porosity, and mechanical properties compared to a market standard for wound healing. So basically, once your scaffold is placed in the wound, it takes up all that exudate to form the hydrogel, and ultimately resulted in complete tissue regeneration, very importantly with scarless skin tissue formation. We also saw improved cytocompatibility, bioactive release, and what we were aiming to achieve was the enhanced or accelerated wound healing with high quality tissue regeneration as observed histologically as well. Another approach which we looked at was essentially a one-step approach, an in situ conjugation co-fabrication approach, also employing here 3D bioplotting for a nanostructured bone scaffold. So the inclusion of the nano additions here was your nanostructuring by your silica, nanoclay, or hydroxyapatite. And this was then used as a bioink. So the single step approach employed here was by combining your anionic and cationic hydrogel together with your nanostructures as a single bioink, which was then 3D printed, and then forming that polyelectrolyte complex by the interaction of course, your anionic sodium alginate and your cationic PEI. SEM visualization shows basically that you have a structure that replicates the required surface roughness required in an extracellular matrix environment for promoting your cellular adhesion. MRI imaging showed the hydration of the scaffold, and biotester imagery for the physico-mechanical analysis showed the essential scaffold strength that was required, obviously because it's going to be implanted into a bone site. Very important is the observed cellular differentiation, adherence, and growth of your osteoblasts following incubation with your scaffold. So basically, after 1 day, the cells already demonstrated adherence and growth to the bioprinted scaffold, and from the 3rd day, you can see that the cells are forming colonies, which increased greatly in density at day 7 already. So basically, require progressive approaches for fine-tuning the mechanical properties of these bioplotted scaffolds. And one project we looked at specifically focused on chemical modification of the physico-mechanical properties of the bioplotted scaffold. So various chemical modifications were undertaken with functionalization of these scaffolds by, for example, aminolysis or hydrolysis, and then nano enhancement was by the incorporation of nano strengthening agents for enhanced physical mechanics of these scaffolds. And then I'm just going to touch a bit on this, but Prof. Kumar will expand on this in his in vivo testing approaches in terms of 3D printed and artificial ECM for potential neuroregeneration. As we know, regeneration capacity is insufficient because basically once you have nerve damage, that's it. You know, you have incomplete repair and poor ability to regain function. And there's to date no successful therapeutic scaffold for regeneration of damaged nerves. So what was proposed here was a 3D printed self-assembled biomaterial extracellular matrix for nerve regeneration, combining a polymer which is thermoresponsive and UV cross-linkable with a peptide to create a peptide hydrogel. And this was also a 3D printable system. So basically, we were creating an enhanced extracellular matrix structure to promote the adhesion of your neural cells, promoting neural regeneration. Okay, so in terms of where we are heading in terms of tissue engineering, as well as its convergence with nanotechnology, 3D printing is a growing field, but the largest challenge here is balancing all the performance objectives to create successful scaffold that integrates all the properties that are required within the areas of the biological and physical complexity of your wound healing processes. So a large amount of the current research has shown advancement in one direction. For example, we're seeing a lot of scaffolds that are showing very good biocompatibility, but results in terms of mechanical strength are still not what we want. So we need to integrate appropriate measures to create scaffolds that are biomimicking the ECM completely. And this would need to incorporate scaffolds where they have appropriate porosity to improve cell viability, and getting materials that closely mimic the properties of natural tissue. What we need is more research into novel nanostructured bioinks. And we're also looking into nanostructured scaffolds for gene delivery for actually enhancing the tissue regenerative phase. We're looking at this, for example, in ocular tissues. So further advancement of 3D bioprinting of organs, just a snapshot into the future. Experts are projecting that science is less than 20 years away from developing a fully functioning 3D printed heart. Currently, the 3D printed heart is still challenged, obviously, by the intricate nature of its vasculature. So I'm going to end my presentation there, and thank you to everyone for your attention. Thank you, Prof. Archana, so much for inviting me as a panelist. Thank you again to my team. Prof. Chinara, thank you for the introduction. Prof. Kumar, thank you for all your help with all this work. And to all our postgraduates and postdocs involved in this work. I'll hand over then and take questions in the final session.
Thank you, Lisa. Thank you for that interesting and very complex presentation, as well as elegantly presented. Colleagues, I think that the last two presentations we heard just highlights, the complexity of nanotech and where actually we're heading to. And on the African continent, we do have the capacity and innovation in terms of the science, in the science that needs to be developed. But what our focus also needs to be is how do we get those lower hanging fruits also out? So the continuum that I shared in my introduction, where we take the trajectory from basic oral delivery of nanoparticles, I think on the continent there's pockets of excellence that move beyond that now, where we're no longer talking about nanoparticles and oral delivery. It's more the complex issues. But what's not coming in, not aligned to what we actually need to achieve, is the regulatory, the product development, the commercial side, the partnering, the contextually relevancy of the technologies. And therefore, as Lisa has mentioned there, the wound healing application in Africa, how do we take tissue engineering that is global and contextualize it to what's the needs of our continent, and at the same time, not reducing the scientific impetus that needs to be there, but at the same time, moving towards a commercial or clinical application of that. The next presenter is Prof. Pradeep Kumar, also from our team at the Virts Advanced Drug Delivery Platform. And he's going to be sharing with you some of the ex vivo and in vivo techniques that we use in the technologies that Lisa has highlighted, as well as some other technologies that's on the way, where we're merging nanotechnology with tissue engineering. Thank you, Pradeep. It's all for you.
Thank you, Prof. Let me share my slide. I hope it is visible to everyone.
Yes, thank you.
Okay. So yeah, thank you, Prof. Chinara and Prof. Archana for giving me the opportunity to present, and to Unica as well. So I will not be presenting too much on ex vivo because I think Lisa already covered, and And the benefit of presenting at the end of a webinar is that you don't have to deal with too much introduction. So I will just start straight away. And so there's a disclaimer that all information presented here is not published as yet, and it's approved by the Animal Research Ethics Committee at WITS, and which I was a member for 2 years, and now Lisa is a member for another 2 years. So I will be discussing 4 different models straight away: full thickness burn model, as well as the superficial one, and then two of the neurotrauma models. So in a sense, the center of all these models that I'm presenting today is nanofibrous mats, or what we call as nanofibers. So they are usually electrospun, and there are other ways to make nanofibers as well, but I will be discussing the electrospun nanofibers. So as mentioned by the colleagues from Mauritius, as well as from Lisa and yourself, so nanofibrous mats are crucial for tissue engineering aspects in terms of nano scale. So we all know that nanoscale is something where we have at least one dimension less than 100 nanometers, and nanofibers usually fall into this category. So we do a lot of image processing of nanofibers. So you can see in the front, so we have nanofibers, and then we do a porosity analysis using Imagear software. And then you can check the size of the nanoparticles using the same diameter J plugin. So, for example, the previous one that I'm showing here, you can see the most of the nanoparticles are in the size range of 50 nanometers, but few of them can go up to 300 nanometers. But mostly it is in the range of less than 100 nanometers, and we can call them as nanofibers. Again, orientation is very important. So we use orientation J for checking the orientation. You can see from the 0 to -90 and 0 to 90, they are mirror images. This means this mimics the perfect ECM architecture of the natural tissue matrix. So everyone is saying these days that you have to have an aligned structure. Some people say a non-aligned structure will work, but I always mention this thing that if we have a mix of both alignment and some, nature is not perfect. So if we have some imperfections with some alignments, and then we can get a very nice ECM mimicking scaffold. So the first one going forward is from Nema Mayat. So she did a PhD in burn wound dressing, and she used a full thickness burn wound model, where you have to first shave the dorsal region, then anesthetize the rat. You apply some block induction so that there is not a local anesthetic, for example, okay, and then you apply a burn wound induction. In this case, usually a hot stub is used. Sorry, the pictures are a little bit graphic, but I suppose we all are scientists here. Then the outline of burn wound is created, and we cut the piece out, and then it creates a full thickness deep wound. So it is not exactly the same as a typical burn wound where we just burn and we don't take the skin out. So because the skin eventually falls off anyways. So the full thickness deep wound is created by cutting out the skin, and then we apply the nanofibrous mat or the film or a commercial control or a conventional control. I will not be showing many results here because we are in very advanced stages of commercializing this product, but we already have 4 patents in this for this technology under US, Europe, Japan, and China jurisdiction, and I think Aripo as well is under examination at the moment. But I can show you one thing is that the nanofibrous mat and the film, they stick very easily, and they stick, so they are wound. So as soon as they come in contact with wound fluid, they absorb and they stick, they self-adhesive action. The second model is recently done by Dr. Hilary Ndlovu, and so Lisa already discussed the bio platform in her presentation. So I will just be presenting the wound model here. So it was a partially cross-linked, lyophilized, pulverized in situ forming hydrogel using a polyelectrolyte complex, and then it was applied to 4 areas. Okay, so you can say we have 2 rats, and so 1 rat can be used as its own control, as well as two samples can be applied to 1 rat. So it is a very nice model, as uh, as Noshin was mentioning. Actually, yeah, I think it was Noshin was mentioning the 3 R's: reduce. So if you use 2 samples on same rat, you can obviously reduce the number of animals. So these were the results. So we found regeneration of scarless skin tissues after 15 days of wound healing, and as well as we reported accelerated tissue remodeling and wound healing at early stages. So it's very important. So at the end of the 14 days, you can see all 4 of them have healed, but you can see that the real difference lies when it is day number 3 or day number 7. So by the day number 7, in case of, you can see the last one, the alginate RGD chitosan and the transferolic acid scaffold. In this case, the day 7, the complete healing can be seen. It can be so we also further we also image process this one to get the real picture, and see and to differentiate the how the wound is remodeling, as well as the tissue is healing at the as the time goes. So the third one is okay. After the wound dressing, I will go into the neurotrauma. So recently graduated Dr. Gillian Mahomane. So she already graduated this year, and she made a very complex scaffold, 3-in-1 3D multi-composite nano micro macroporous scaffold, which included a hydrogel, a cryogel, and nanofiber. So I know Archana mentioned something similar, which they are also working on as a composite structure. So this is a nice synergy. Okay, so we never discussed. We can see like how this field is now growing in Africa, where 2 different labs with no similar discussion are working on a similar concept, so which is very encouraging. So this is our brain injury model, where we already have a rat fixed in using a stereotaxic frame, and we check the coordinates, make a 5 millimeter incision, and then take the tissue out and implant the scaffold. So that's how it works. So it is a very complex process, and so our surgeon works very closely with us. Neurosurgeons work very closely with us, and they usually help us with all these studies. So you can see in this one, if you see the bottom on the scaffold part, it filled the scaffold site. Okay, so the scaffold filled the tissue site, maintained tissue scaffold intact, and remain intact within with interconnected pores when we took it out, as well as was progressively infiltrated with cells. So you can see, like it's very difficult to test this infiltration of cells, as well as interconnected pores, and most importantly, the tissue scaffold contact in vitro studies or even ex vivo studies. So somehow we need in vivo somewhere to test all these to test all these new technologies. Okay, so all this work is unpublished, and we will be publishing these very soon. But the students have graduated recently. So she also did some behavioral tests, where we have cylinder tests for limb use symmetry, tactile sensitivity, neurological severity score. So it's not only for neuro, we all know that neuro is not only about tissue regeneration, the functional behavior is also very important. So if the tissue is meant together, but the thing is the rat is not moving, or is not doing the right function, then there is no point of doing the animal studies as well as the technology. So it could be biocompatible, biodegradable, and everything is possible, but unless we have the final performance analysis, we cannot be so sure. The last one I will mention is the spinal cord injury model, which is our category 6 study model, which is the highest category that a study can go into, and it's very critical to be assessed by control committees as well as the screening committee. So this is a spinal cord before injury. You can see the cortical neurons, interneurons, sensory neurons, motor and central canal, they're all intact. So everything is working perfectly, and that's how the rat behaves when the rat is perfectly fine and before the spinal cord injury. They can stand and they can move around, and they can lift the tail as well. In this, so when we, so if we take a small piece out of the spinal cord and disrupting all these neurological functions, then motor neurons will stop working, for example, and you can see the arrows. There's a block in between now, so there's a cut or block in between, and we cannot now see any motor function in the hind limb. So the rat has got no hind limb function, and the tail cannot move because of spinal cord injury at T8 to T10. It is very disturbing images and everything, but you know, for science, I think we have to make a compromise. So after you put a scaffold, so I'm showing here an aligned scaffold with the nanofiber system, and then it can go into this spinal cord, and the motor neurons can, if it is melded together, and we have sensory or cortical neurons, which are on the side of the spinal cord, not the central canal. I'm not talking about central canal. So the cortical neurons and the sensory neurons can, the blue and the pink ones can grow, so we can always have some motor function coming back. So you can see the rat on day 0, 4, 7, 21, and 28, and the spinal cord is it's not 100%, if at least 90% it is regenerated and it's joined together. And you can see the rat can stand. So I will show you this one. So this is the rat after 28 days. So there is some kind of some small amount of limping in the rat. The tail is now functional. It can and so it get imbalanced a little bit. Yes. So yeah. So that was my presentation for today. I always say while this rat is enjoying itself, we'll say in vivo is still important to get into the clinical and commercial space, and before we even like no behavioral, the most important one is behavioral function, as well as if you go to clinical trials, we need preclinical data, and if it is done at the researcher stage, is always very good. So thank you everyone, and I dedicate this, all research is to because of Prof. Penes Pillay, who sadly passed away last year, and these are our funders. Thank you, Prof. Chunara, and thank you, Prof. Archana.
Thank you, Pradeep, for that presentation. Colleagues, just a comment on that. I mean, we chose to show the the technology, the very complex ones, just to highlight that with nanomedicine, it's not about working in silos, and that's not going to get us anywhere. We to put the projects of these natures together, as Prof. Kumar and Dutoy has just shown us. It takes a lot of work with collaborations with clinicians as well, to make sure that the research strategy is aligned to what's the needs of the market. So it's a needs-based research area. So that's why obviously the alignment to the wound healing application, spinal cord injury, we know, I mean, in South Africa, at least, we know we are very violence and assault and crime is quite big. So spinal cord injury is quite a big thing for us as well, and contextually. So we try to see how we actually, how complex these biological systems are, and that's for the need for us to collaborate more on the continent. Archana, I'm going to hand it over back to you now to introduce our next speaker. And then I think for the rest of the program, we'll take it from there. Thank you.
Yes, thank you. Thank you. And this is a very nice introduction to Fanny's talk. Dr. Fanny Gimier is actually a vet. She works at the Siwa Center. She's responsible of the animal house, and she's also the president of the Ethical Clearance Committee of Réunion Island. Therefore, it is really why we kept her at the end, because she is on that side, that is the ethics side. And this is really where we need, as you said, we need different expertise coming together for this area of nanotech. Fanny, the floor is yours.
Thank you. Thank you. I would like to thank Archana for this invitation and for giving me the opportunity to present our research platform, as well as our skills, in particular in studies related to nanotechnologies. My presentation will be in English. I apologize in advance for any mistakes I could make or the mistakes of pronunciation. But maybe I will switch to French for questions. I'm going to share my presentation. So I'm Fanny Gimier, I'm a research vet. I'm also the manager at the animal facility at Sirois and chair of the ethics committee in animal research of La Réunion. First, I would like to introduce the research platform of Sirois, which hosts a great diversity of research teams and scientific activities. Then I will talk about ethics and the use of animals in research, including regulatory requirements. At last, I will talk about our expertise regarding in vivo techniques and models. Sira is a unique technology platform created in 2008 on La Réunion. Its management system is certified with ISO 9001 and AFNO quality standards and aims at developing different fields of activity. We can distinguish 4 main fields of activity. First, Sira can produce a drug, the 18-FDG, thanks to its cyclotron. 18-FDG is a short-lived radio tracer used for cancer imaging by PET scan. This local production avoids medical evacuations to mainland France and promotes equal health care between La Réunion and mainland France. Second, Sira has several research units working on different fields. These different teams collaborate with different partners in Indian Ocean area and also provide services for public or private companies. The BioAir units work on plant extraction and phytochemical analysis. The RIPA radiochemistry and small animal imaging unit is specialized in radio labeling and studies the biodistribution of radio-labeled molecules in small animals like mice, rats, hamsters, and rabbits. For this purpose, Sira has acquired a tri-modality imaging system, including PET, SPECT, and CT scan. The analytical chemistry unit has an expertise in the chemical characterization of volatile compounds. At last, the animal research unit has different facilities and works on different animal models that I will further detail. The Sira platform also hosts different research teams working in private or academic institutes like INSERM, CNRS, CIRAD, University of La Réunion. The 2 main research teams are UMR-D3, this one, which studies the vascular issues of diabetes and therapeutic leads using molecules from the biodiversity of Indian Ocean area, plants, fruit, algae. And UMR-PMIT is specialized in the physiological and ecological aspects of infectious disease. The team studies the impact of the ecosystem on disease transmission and their immunophysiological consequences on human and animal health. At last, Sira supports innovation thanks to the CBTech business incubator, which provides facilities and services to young companies innovating in biotechnologies. The Sira platform has the only animal facility in southwestern Indian Ocean area that meets the regulatory requirements established by the 2010 European Directive. This facility allows the practice of surgery and microsurgery, so we can develop custom surgical models. As mentioned earlier, There also is an imaging system with SPECT-CT modalities. Besides our involvement in animal research, we had the only ethics committee on animal use in research of La Réunion, approved by the Ministry of Research. This committee monitors the use of animals in scientific research studies, including laboratory and wild animals like bats, birds, marine turtles, or dolphins. We also dedicate a significant amount of time to train people like technicians or researchers so they can use animals in research with respect to the current legislation. What about the regulatory requirement regarding ethics? The 2010 European Directive is the latest update of the legislation concerning the protection of animals in research. Any researcher designing scientific studies involving the use of animals, vertebrates or cephalopods, is required to obtain an authorization from the Ethics Committee. Also, people using animals for scientific purpose are required to pass a specific training and obtain an official certificate. People designing experiments, researchers, and people performing experiments, technicians, follow different trainings. People performing surgical procedures also have to obtain a certificate by following a specific regulatory training. Continuous learning on animal welfare is also required and inspected by the government. The principles of ethics in animal research can be summarized in 3 R described by 2 British researchers in 1950. 3 R stands for replace. We have to use methods avoiding or replacing the use of animals when possible. For example, in vitro and ex vivo techniques can be used as a replacement. The requirement of animal in each study has to be justified. Reduce. We have to minimize the number of animals per study while maximizing the information per animal and ensuring robust and reproducible findings. This can be done by improving the design of experiments, such as using adequate statistics or mastering technical skills to avoid mortality. And refine. Experimental procedure has to be improved to promote animal welfare by minimizing the pain, stress, and distress of animals. This can be done by enriched housing allowing animals to express their natural behavior, the use of appropriate anesthetics and analgesics, the control and limitation of animals' discomfort, etc. The Ethics Committee evaluates every scientific project involving the use of animals. The aims of the evaluation are to check the clarity of the scientific objectives and the rightful requirement of animals, the respect of animal welfare, and the optimization of the use of animals, the techniques and methods used, choice of animal species, number of animals, anesthesia, analgesia, endpoints, etc. Now I will present the in vivo models we are developing at CIRA. The main fields of activity are the assessment of activity, efficiency, and toxicity of active compounds. These studies are conducted after the characterization of these compounds on cells and tissue models. The production of antibodies involved in specific disease, in particular infectious disease. The development of animal models for cancer research. We mainly use immunodepressed animals on which we can transplant human tumoral cell lines and process to diagnostic or therapeutic research. For this purpose, we use the trimodality imaging system. And the development of specific surgical models using microsurgery procedures such as organ transplants. This can be done on the surgical platform located in the animal facility, which includes surgical microscopes. At last, I'm going to detail the in vivo models we have used in the field of nanotechnologies. We can assess the biocompatibility of innovative biomaterials. Once these biomaterials have been characterized in vitro, their biocompatibility can be checked by implanting them subcutaneously for a few weeks, 2 to 4 weeks. Then we can take the implant back and assess its physicochemical and biological properties, for example, its degradation. By using this model, we can reuse the same animal for another study, given that the implanted scaffold needs to be in contact with just a little piece of skin. Here we can see a skin wound healing model. For this purpose, wounds are done on rat skin with the help of biopsy punches. 4 wounds can be done on one animal, one rat. A splint is placed and fixed around the wound to avoid wound healing by contraction of skin muscles, which is the main physiological wound healing system in rats and rodents. Thanks to this model, we can assess the healing properties of different scaffolds. These pictures represent the evolution of the wound healing process by using different scaffolds, and we can see that the healing process is different depending on the scaffold. Histological analyses are currently in process, and ELISAs will be performed to analyze specific protein contents on skin tissue homogeneity. We are currently studying the healing properties of innovative scaffolds on bone regeneration. For this, we use a model of calvaria defect on Wistar rats thanks to a drill. The healing process is monitored by using the sodium fluoride 18 radio tracer, or NAF radio tracer, and TEP scan imaging. This radio tracer can visualize osteoblastic activity. The advantage of this imaging technique is that we can see the colonization of osteoblasts in the scaffolds that we will integrate into the bone wound and without mortality. Besides an animal experiment, we have developed other technical skills in cell culture, including primary cell culture, in histology, histochemistry, and immunohistochemistry, the extraction and titration of protein and nucleic acid from cells and tissues by using molecular biology techniques. To conclude, on La Réunion, animal experimentation is regulated and controlled by national and European authorities. The use of animals in scientific research has to be carefully decided and justified. All studies involving the use of animals are evaluated by ethics committee, approved by the government. To minimize the use of animals, the development of alternative methods is encouraged. And the protection of animals is currently a hot topic, and not only in the research field. Thank you for your attention.
Thank you very much, Fanny. And this was a very proper end of the lectures because it's the ethics. And I can tell you that we have got lots of discussion with Fanny before we go for any in vivo experiments, because she makes sure that we are doing it according to ethics and according to the 3 principles. Now we will be taking questions. The Samuel is not with us. He's been called for an urgency, and I think you can send your question on the chat for the attendees, because we can't, I don't know if you can speak, because the way he has divided panelists and attendees, but if you have questions, can you send them on the chat and we will take the questions. Yaya, is there anything you want to add before
No,
we open for discussion on other things, if there are questions for the
no, nothing from my side. Just to say that it's a wonderful package of research that we've seen in terms of the complexity, and that's what I just want to highlight is that there is the lower hanging fruit for Africa, but we also have the capacity to have the higher level of science to happen on the continent, and in some way or somehow we need to make that turn it into impact. That's all my comment for now. I look forward to the questions from attendees via the chat, and I agree with it. Thanks, Archana.
Okay. Therefore, if you have any questions, you can send them on the chat and we will take the questions. We will wait for the questions to come in. We have around 15 minutes to finish the webinar. We can take questions. Students among the attendees, if you have questions, you can send them on the chat. Yeah, by the time that we've got questions, you had the slide for the MSc, your last slide,
yes,
where you've put the MSc Nanotech.
Yes, for sure. Do you want me to share that, Archana?
Yes, yes. Maybe you could share that last slide so that this group could know what is going on in terms of that MSc Nanotech. I think I saw Géraldine Dantel, who is the consultant for that, is on the list of attendees, but I don't know if we can upgrade her. I think only Samuel could do that. So that if she had, I can see we have a hand raised. Yes, Géraldine has raised a hand, but I don't know if we can upgrade her so that she can intervene, because we can't do that, I think. Let me see. View options. Yeah, maybe you can talk a bit about this slide, because Géraldine has raised a hand, but we can't, I'm not able to.
Okay, thank you, Ashley.
Okay.
I think where this webinar was born from is our discussions that we were having, on the creating of a Pan-African nanotechnology online master's program for more directed training and capacity development on the continent, so that we could then take these complex research and pockets of excellence of research that we have on the continent. I mean, I'm talking about, the tissue engineering, the wound healing, the classical nanoparticles for TB, malaria, HIV, your nanoliposomes, solid lipid nanoparticles, targeting systems, whatever we may have within our research teams and groups. And how do we actually target that so that we could get towards that, the goal where we talked about, commercializing nanomedicine, in particular for Africa, and having the revolution of nanotechnology really take action in Africa. So some of the things that we looked at, in terms of curriculum content, it needs to be a needs-based education. So the gaps must be focused on things like, dispelling all the controversies about nanotechnology, whether it's safe, not safe. There's still lots of that kind of, people sitting on two sides of the fence. And what a needs-based education, and part of this online master's program or any other form or shape in which a partnership is happens within the Southern African region, is to fill these gaps. So first is to dispel controversy, mainly around the occupational health, safety and environment, the ethical, legal, social, regulatory doubts that are there. And the reason these doubts happen is because there's a lack of education. If people are educated about what nanotech can do, what it, or not, you see, the trick is not to overpromise. And I think that is some of the things that are happening on the continent, is where, us as some scientists and, leading scientists also, if you overpromise on what you can achieve with a certain technology, what it actually does is that if you do not attain that commercial product within a certain amount of years, eventually the public, as well as the politicians and the funders, they start getting demotivated, whether this technology actually works or not. So I think taking it also in baby steps to make sure the fundamentals are in place first. We understand the biological systems very well. We understand the nanostructures very well, which I think we all are very good in understanding the nanostructures. But how these structures interact with the body, I think we still have a lot of fundamental questions that need to be asked before we can claim or put out that big claim that, this is the next revolutionary blockbuster product that's going to heal the whole of Africa using nanomedicine. And we need to guard against that and find that balance. So education is key, to dispel these doubts. And I think the doubts are created because of, our zeal for wanting to put out nanomedicine very quick. The second thing is understanding the mindsets or the intellectual culture that give rise to the contradiction. The slowing growth into clinical practice is like, what is the mindset or the intellectual culture on this? You have everybody working on, it speaks to my next point. We have lots of people working on nanoparticles, from pharmacists, biologists, physicists, chemists, engineers, and all of them call themselves nanotechnologists. I mean, we all call ourselves nanotechnologists because we are producing nano devices. But in terms of the background training, our mindset and intellectual culture, how does it exist within each of these groupings? The engineers might think of nanomedicine in a different way, similarly with the chemists, the physicists, the biologists, and the pharmacists. But we all claim or wanting to produce nanomedicines, or we all nanotechnologists. And I think that communication between each of these groups, whether you're an engineer, chemist, biologist, pharmacist, it's all required. And I'll add even the clinicians to this lot. That's required to make sure that the nanomedicine actually makes an impact. Because of we're dealing with particles where we always say the physical-chemical and physical-mechanical principles no longer hold, the fundamentals no longer hold because we're at such a small level. So same things, the relationships where we work in silos can no longer hold if we want to make sure we have nanomedicines that can reach the market of patients. So physicists, chemists, engineers are trained to mainly deal with the physical or machine systems, as we call it. And these systems are largely predictable. They are mathematically stable systems. We can use mathematical models to stabilize them. When it comes to the biological system or the human body, the body itself adapts to changes. And no matter how much of mathematical algorithms you develop, you're dealing with an unknown. I mean, I always say, a bacteria has a brain of its own, a virus has a brain of its own. We've seen that with the pandemic, in that we have, mutations and variations. And similarly with the body, the behavior is very hard to predict. So although we can design very stable nanoparticles, as material using mathematical models, but when we apply these nanoparticles to a living system, whether it be a cell, a virus, genetic material of the body, the rules don't hold. And I think that's where the fundamentals are getting lost. And having this kind of a training base or capacity development in the online, within Africa, where we can train our people to actually get to the bottom of the understanding the biological aspects as well as the nano materials aspects, that's where we can bring nanomedicines to the fore. So collaboration is definitely needed, interprofessional approach, interprofessional education. I mean, if you look at the pandemic that we're currently in, deeper connectivity is needed, agility and networking. And that's what the pandemic has taught us, that we need to cross the borders to solve global solutions, and we can't be working in these silos. So deeper connectivity around the fundamentals, deeper connectivity, and then not much more on the so-called commercial side. We understand what's required commercially, but have we actually worked out what is the hurdles, the regulatory hurdles, and all of these things that science brings to the fore to solve these problems? And I think that's where collaboration is needed. So the area of focus, I mean, the first generation nanomedicines have been in clinical practice now for more than 20 years. Well, we all know how to make nanoparticles. We know how to deliver drugs with nanoparticles for, TB, HIV, malaria, cancer. A lot of products in the cancer space has been already on the market, and the literature has doubled. With more than 50% of these, papers in the last 5 years in these areas of nanomedicines. But I think the second generation of research that needs to happen is to look more deeper into the biological interactions. And here we're talking about the immunotherapies. How does the immune system react to these particles coming into the body? The bioresponsivity, how does the body respond? Because we're adding a foreign material to the body. I mean, we know how drugs respond, but how does nanoparticles respond and cell interactions? And I think understanding the fundamentals and building capacity in these three areas are going to allow us to get to understand the fundamentals better and get to the second generation of products that can come from Africa. And also, I think we have good snippets of products that can be developed from the first generation of nanomedicines that have gone past already. But understanding these is where the world is heading, where I think Africa should not get left behind. And certainly from a scientific point of view, we do have pockets of excellence, but it's the collaboration and the access to facilities, access to world-class research labs will allow us to put us on the map. And also, the commercial case for Africa needs to be made higher up, in that how do we get the like partnering with UNECA and other bodies? How do we get the commercial case strengthened so that the big farmers, all the small farmers, all the people with vision can see where these technologies can be made a reality in Africa. Facilities and personnel are critical. I mean, like we've spoken about some research at our unit. We have things like a nano assembler, 3D bioplotter, Elastosense Bio. These are all very expensive equipment, and some of them are like the first lab in the world to have all three within one laboratory. And that is existing in Africa. And yet we don't have, the question keeps arising, why don't we have why don't we have our own vaccine development? So there is certainly a gap, from higher up, how do we showcase, exhibit, and drive nanomedicines to get to the end result. And duplication is also a big problem across the continent. Everybody wants to set up the next large research institute. Everybody wants to have the same instrument in their labs, advanced instruments. And this is actually draining resources rather than working together. And it slows the progress of all of us together. So, having 10 different nanomedicine research institutes on the continent is not going to help us. I mean, investment of rather having PIs focused on very specific research that has good science that can be commercialized. Money will be well spent on that area, and training and capacity should be the big focus now. So rather have a big research center instead of with a lot of fancy equipment and materials, but rather have lots of fancy people that can be trained. And then those people will go off to set up their, whether it's big labs or mini labs, but doing good science that can be commercialized. And then public-private partnerships are very important. That's for third stream income, for new workshops, short courses, IP and licensing programs on the continent. How do we drive those? We need the private sector on board for that. And entrepreneurship and innovation is very important. I mean, at WADEP and at the Department of Pharmacy at WITS, we have a program we call Farm Apprentice Program, where all our postgrad and undergrad students are given an entrepreneurship spin embedded into the curriculum. So when you're doing that, background curriculum, undergrad or postgrad, you understand the importance of your research towards entrepreneurship. It's not only self-serving. Entrepreneurship obviously helps society and plows out more funds back into the economy. So that's in a nutshell, Archana, in terms of this slide and where this webinar was born from and where I think we should work together to actually move toward. Thank you for that.
Yes, thank you. Thanks a lot, because in fact, what Yaya's presented to you, we started the group that was involved in that. We started discussing since last year, because we identified really a training and having people with different expertise as a must for the African continent. Geraldine is here. She has been able to have upgraded her as panelist. Before I pass on to Geraldine, because she's the one looking at the generic nanotechnology program for Africa. We have two questions on the chat. It's from Dr. Maheshwar Gupat. Dr. Maheshwar Gupat, he's a medical doctor, and he has asked, does research on use of nanomaterials for drug delivery require ethic clearance, or do we only need proof of concept in vitro studies? That is, we are using drugs, known drugs in our nano devices. Therefore, what type of approval do we need? Do we need ethical clearance for the whole thing, or just proof of concept? Maybe Yaya or Pradeep can take this question.
Thank you, Archana. Can you hear me? Yes.
Yes, yes, can hear you.
Does research on nanomaterials for drug delivery require ethic clearance, or do we only need proof of concept in vitro studies only?
Yes.
So I think the ethical clearance comes in when you're doing your preclinical in vivo animal studies. Animal ethics is involved there. And then if that progresses any further or beyond animal studies, you will then need an ethics clearance for the human studies or the pilot human bio studies. But just to undertake nanomaterials research in drug delivery, you as a scientist, I mean, you can go ahead with that as long as you are using, obviously you're not using materials that are born from animals or humans to make those nanomaterials. But otherwise, if it's classical polymeric drug delivery, you obviously can go ahead without ethical clearance. It's just when the animals and humans are involved.
Thank you, Yaya. There's a question for Noshin on the chat. Noshin, can you answer the question? It's about the spheroids.
Yes, so the question is about how do we control the size of the spheroids, and can this type of system be applied for bone tissue engineering? So the answer is yes, you can control the size of the spheroids. And how do you control the size of the spheroids is by keeping the number of cells in the spheroids constant. There are various methods that you can use to fabricate spheroids. For example, one of the methods is using the hanging drop method. In this method, what you do is you're going to count the number of cells, and then you're going to determine exactly how many cells there will be in one drop. You're then going to place single droplets on the inverted lid of a Petri dish. And then this lid is then inverted over a Petri dish. So by ensuring that you have exact number of cells in each droplet, you can control the size of the spheroids. For example, if you want a larger size spheroid, you're going to include more cells when you seed the drop on the lid of the Petri dish. And the second part of the question was, can this type of system be applied for bone tissue engineering? Yes, you can apply spheroid system for bone tissue engineering. In fact, there are many different spheroid systems that you can use for bone tissue regeneration. You can have, what I forgot to mention in my presentation is that the spheroids, they can consist of a single cell type, or they can also consist of 2 or more different cell types. So if you're looking into bone tissue engineering applications, you can opt, for example, in making spheroids which consist of only pre-osteoblasts, for example. Or you could use spheroids which consist of stem cells in combination with endothelial cells, where you're going to provide growth factors to induce the differentiation of the stem cells into bone cells. So I hope this answers the question.
Okay, thank you, Noshin. Geraldine, can you talk now?
Yes, can you hear me?
Yes, we can hear you.
Yeah.
Yaya has shown to us the slide about how the discussion started really about the Pan-African Masters Program, and you are working on that. Maybe you can give the attendees an overview of what is being done on that.
Yes, thank you, Archana, for giving me the opportunity to speak now. So maybe just a brief introduction. So my name is Géraldine Dantel. I'm a researcher in nanotechnology at the French Scientific National Center, the CNRS in France. And I'm also a consultant under the guidance of UNECA to design a generic BSc and an MSc curriculum in material science and nanotechnology. So I've been working on this curriculum for a few months now. And I think, as you mentioned, the key point is really to answer the question how to get more students educated in the field of material science and nanotechnology in order to build a larger pool of nanotech scientists that can then further teach to the new generation of students and also bring innovation to African countries. From the work I've been doing on this generic curriculum, I would say that there are different ways to implement this program, depending on the countries, depending on the already existing programs, and the will to really implement either a new program or to work on already existing programs. So I would say I would say depends on the country. I would say it depends on the resources that are available, either infrastructures or teachers, researchers that can teach these courses. But just to take an example, the one way to form or to have students that are more informed about nanotechnology could be to introduce a few courses at the BSc level, for example, in order to give those students a larger knowledge of what nanotechnology is and how it can be implemented in the everyday life and in some innovative products. So I would say that could be a first way to go, and that could be implemented rather rapidly on already existing curricula, either in chemistry or physics. You can imagine that you add a few courses that would then allow the student to have this larger knowledge about nanotechnologies. Regarding a more structured curriculum, I agree with what was said, in the sense that there is no point in developing in each country a nanotechnology centers, which are very expensive, require very expensive pieces of equipment. But I would suggest to define some specific tracks, and you mentioned them during this webinar, with 4 main domains that have very strong consequences on African countries. That is energy, water, agriculture, and health. And to design some specific education tracks that would be targeting these 4 domains. And I would imagine to create a network between universities where you would have students that could choose one or the other domain to be specialized, and that would develop some specific skills in one of these domains according to their wills. And I would think that this kind of structuration would be very cost effective and interesting for the development of nanotechnology in Africa. So maybe that's what I can say now very rapidly as a kind of summary of what we've been doing over the last weeks. I don't know.
Yes, thank you, Geraldine. It's you know, so that people can know what is coming in. That is really the vision for nanotechnology in Africa. It's not a one-off. That is, this effort has started last year, and we want to keep this going so that we can reach really momentum with the program, the training program, with putting the labs together so that we can use the facilities in different labs and the expertise in different labs. I think I'm just checking if we have any new question. I think we answered the second question also of Dr. Ragupat, because he gave examples of some of the, he talked about hormonal implant depot for pregnancy prevention or blood forming agent. I think he was talking about the ethical clearance, and he gave examples of them. And if there is no other question, is there any other point the panelists would like to make before we conclude the session? Anybody wants to add something before we close the session? Yaya, yes?
Thank you, Archana, for the opportunity again. Yeah, no, so I just think, you know, it's important that we drive the training and capacity development issue. The way I see it in Africa at the moment, the pyramid for capacity development is actually inverted. So at the top, we have lots of innovation, good science, pockets of excellence that's going on. But when you talk about people, there's very few specialists in these areas. So if you want to commercialize, we shouldn't forget that, you know, once a technology is transferred to a company, that company or institution will need all the support it needs, the consultancy support it needs to translate or further develop that into a product. And, you know, if it's just a handful of scientists developing things on the top, and then when it filters down to the commercial side, you don't have people in the market that they can employ who are experts in that technology. So you're actually reaching a bottleneck very quick. And having a good training program, as Geraldine has mentioned as well, where a virtual program where we could feed into developing capacity in this area will be great. I mean, we are very strong at doing research on solutions that pose more problems. Solutions that you can identify more problems. If another lack of a word, solutions in search of a problem. But rather, we should look at technologies that realistically address the clinical problems that are out there. Thank you.
I fully agree with you, because sometimes when when we are working, we are looking for new people to join the group, and you're struggling to get the expertise that you want, and you yourself, you have to train the person so that you get the expertise that you require for the work that you want to do. And then once you train the person, you don't want the person to go, because it becomes difficult to recruit another person and retrain that person. I fully agree with you. I don't know if you have that problem, but this is getting the human capacity is a crucial part. And I would like to also conclude by saying that whatever we have discussed today, we will probably publish it in a specific journal, and Yaya will be contacting you for that, and so that we keep the momentum going. I can see there is I don't know if there's a third question. Yeah, there is a question on the chat. Geraldine, can you see the question? It's about the master's program. Geraldine, if you're still here, can you see the question?
Yes.
It's about the nanotechnology master's program. And there is somebody from the University of Western Cape that coordinates already an MSc nanoscience program. I think you have an idea what is going on the continent from the presentation you made last time at the UNICA meeting.
Yes, so indeed, well, the idea was really to build up a generic curriculum that then can be applied in different universities and adjusted. And the idea is not to compete with already existing MSc programs. There are a few, indeed, MSc programs that are very well structured over some African countries. But the idea was really to propose a generic program that could be implemented in other universities, and that should be flexible enough so that universities can adjust it as they want, and depending, as I said, on the infrastructure and the human resources and so on. So of course, there are a few programs that exist already, and that's great, honestly. The idea is more to see how those courses or nanotechnology in general can be can be taught in a broader way in other countries and in other universities. So if some program exists already, and we have taken that into account already, I mean, that's great. The idea is not to to compete with those programs. The idea is really to open up nanotechnology programs in other university. And as Archana said, there is a group of people that have been working on how to create more, to increase a network about nanotechnology, so that skills can be spread out over African countries. I don't know if I answered the question.
Yeah. Yes, maybe we could also say that the way that the program is being designed is that if there is an existing program, they could plug in modules from the program, the generic program, and add to the existing program.
Yeah, exactly. Yeah.
Yeah. Then you can strengthen the existing program. And I think with this, we will end the session. It was really a pleasure listening to everybody and having taking the question. And I'm very happy that Geraldine could also join us. And we will keep contacting you if there are new things coming in. And it's the AMRS who's going to coordinate all the activities in terms of nanotech. Therefore, if there are any upcoming webinar discussion and more information about the MSc program, the generic program that is being mounted, all information will be circulated. And I would like to thank all the panelists, Yaya, thank you very much, Lisa, Pradeep, Fanny, Noshin, Itisha, thank you very much. And we can see that in the region, at least we are starting to put things together. That is, we can use our expertise to do things together. Yaya, would you like to add something to close the session?
No, thank you, Archana. I think it was a great session, and I look forward to the next steps. And obviously, as Dr. Kondi mentioned earlier, it's now the time for action.
Yes, it's time for action. That is, like I've shown in the introduction, that is previously it was just the PI, then it became labs. Now it's really the continental movement. It's time for action. And thank you again, everybody, to the attendees, the panelists, and we keep in touch through email. And thank you.
Thank you, everyone. Bye-bye.
Bye.
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 Thanks. Bye.